Good afternoon to one and all. I, Zaira Uchel,
And I'm Hussein.
we're delighted to welcome you all to the postgraduate session of the 5th Student Research Congress, organized by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by the University of Mumbai, along with our esteemed industry partners, ACG Worldwide and SciTech Center. As C.V. Raman once said, the true value of a scientist lies not in the knowledge they possess, but in their ability to question and imagine the unknown. This competition served as a valuable platform for postgraduate students to present their research and contribute to the advancement of knowledge within their field. Today, we've come together to celebrate the creativity and dedication that fuel research at this exciting event, the oral presentation competition for the postgraduate category. As highlighted by our beloved principal ma'am during the inaugural session, we received an overwhelming response with over 100 entries for the PG abstract. The research committee at BNCP carefully evaluated and selected the best among them. It is important to note that not being selected does not diminish the quality or credibility of any research. Rather, it reflects the constraints of time. As Principal Ma'am rightly said, everyone here is a winner. For all participants, this event offers an opportunity to learn, enhance your knowledge, and grow, contributing to your research journey and future aspirations. I am honored to extend a warm welcome to our esteemed judges whose expertise and insights will undoubtedly inspire and guide us throughout this event.
The judges for the postgraduate category are Dr. Rajkumar, Dr. Manjula, and Dr. Radhika Raheja. I would now like to introduce them. Dr. Rajkumar. Dr. Rajkumar is a seasoned pharmaceutical professional with over 18 years of experience spanning formulation development, biopharmaceutics, clinical pharmacokinetics, and analytical sciences. Currently a professor and head at the Department of Pharmaceutics and Biopharmaceuticals at NIPER, Hajipur. His expertise includes pharmaceutical product development, pharmacokinetic modeling, and life cycle management. Dr. Rajkumar has held pivotal roles in leading pharmaceutical organizations, contributing to the commercialization of over 40 products globally, including complex generics and innovative formulations. With 25 research papers, 11 reviews, 7 granted patents, and over 24 patent applications, including US and EU filings. His work has significantly impacted the pharmaceutical sciences. He has supported more than 160 product developments and played a key role in regulating submissions across major markets. Recognized with multiple awards such as the APTI Pharmaceutics Award and the EPD President Award from Abbott. Dr. Rajkumar is also a Six Sigma Black Belt and holds certifications in advanced pharmaceutical quality and AI and ML applications in pharma. Beyond his technical expertise, he is an active consultant, mentor, and advocate for innovation in pharmaceutical education and practice. We deeply appreciate your time and presence, which have added great value to this event.
Our second judge for the Our second judge for this session is Dr. Manjula. She is a distinguished professor in the Department of Pharmacology at JSS College of Pharmacy, Mysuru. Has an illustrious career spanning over 23 years in teaching and research. Holding a PhD in pharmaceutical sciences from Manipal University, her expertise lies in neuropharmacology, oncology, drug discovery, and cognitive research, with notable contributions to developing therapies for Alzheimer's disease, cancer, diabetes, and metabolic disorders. She has authored over 89 research publications in high-impact journals, presented more than 100 papers at national and international conferences, and has guided over 14 PhD scholars and 55 MPharm PCs. Dr. Manjula has secured cumulative research funding of over 176.47 lakhs from prestigious agencies like DST, AICTE, VGST, and ICMR for innovative projects, including drug discovery using artificial intelligence, chemoprotective agents, and neurodegenerative disease therapies. Our collaborations extend internationally with impactful projects involving institutions like Southern Texas University, USA, and University Technology Mara, Malaysia. Dr. Manjula holds several patents, has organized and participated in numerous scientific events, and has been recognized with several awards for her contributions, making her a leading figure in pharmacological research and academic excellence. Thank you for honoring us with your valuable time and being part of this occasion.
Thank you, Dr. Manjula. I must appreciate your time.
I would also like to introduce Dr. Radhika Raheja. Dr. Radhika Raheja is a seasoned academic with 26 years of experience in pharmaceutical chemistry. Her research focuses on the isolation of phytoconstituents from pharmacologically relevant plants and the synthesis of small molecules. She has contributed significantly to her field, guiding 5 M-Pharm students and collaborating internationally on research projects. She has published research papers in reputed journals, authored several book chapters, and presented papers at conferences. Her recent publications delve into therapeutic peptides, menotropinoids, anti-cancer agents, and phytopharmacology, appearing in high-impact journals. Her dedication to pharmaceutical research and education underscores her impactful career. Thank you for generously dedicating your time and gracing us with your presence at this event.
Request
I would like to take this opportunity to thank all the judges, Dr. Rajkumar, Dr. Manjula, and Dr. Radhika. Thank you very much. I sincerely appreciate your time and your insightful comments will definitely help students participating as well as listening and presenting. So thank you, all the judges, for sparing your valuable time. And I'm sure, and I must say one thing over here is I take this opportunity to just share that today is a birth anniversary of my mentor who has made a profound impact on my career. The entire success of my career, I attribute to him for his guiding. He's Dr. Mukesh Goel, and today he's no more. Today is his birth anniversary. He was my PG guide, PhD guide, as well as the UG guide. Those who know Dr. Mukesh Goel, they will know his contribution in the field of pharmaceutics. So with that, I welcome all the judges and I thank for their presence today and their valuable time. So thank you very much, all the judges. Over to you, students.
Before we begin, I request all participants to make note of instructions carefully. The presenters have to rename themselves as per the allotted codes, if not already done. The presenters are requested to stay online, switch on their camera when their code is called out. Pre-recorded videos will be presented on the screen. Participants cannot reveal their names or any affiliation. Each participant is required to stay alert for their roll call. All the participants should be present for this session. All members from the group are allowed to answer during the question and answer session. The duration for the question and answer session is limited to 5 minutes.
Always remember, the foundation of every successful presentation lies in 4 key pillars: collaboration, innovation, dedication, and preparation. With this note and the permission of our respected judges, let's begin with our oral presentation competition, as all of our participants and audience are eagerly waiting to learn about the new, unique, and interesting ideas that will be presented here. Also, I kindly request the judges to confirm that they have received the scorecard.
Yes, received.
I would call upon code number PG01 to switch on your camera. We will be presenting your video now.
Hello everyone. My Hello everyone. Today my name is today I will be presenting about a topic that is bispec anti-TNF alpha and interleukin 23 antibody has a therapeutic potential for the asthma, its efficacy and the mechanistic study. And my identified code is PG01. As you know, all about means like the severe asthmatic condition, it is characterized by the pulmonary inflammation along with the cough, sneezing, and then the affected individual they literally begs for the breathing. So talking about the epidemiology and the severity of the asthma, it is the second leading cause of the death among the many chronic respiratory disorders, and around 5 lakh fatalities have been reported from all over the world. Talking about the India, it affects nearly 30% in India, and around 2 lakh deaths have been reported every year. And India contributes about 43% of global asthma related deaths. And talking about the future perspective for the global asthma market size, so by end of the year 2032, it has been reported that the global asthma market size will be around 40 US billion dollars. So now talking about my hypothesis. So whenever any antigenic stimulant or any pollutant, it comes in contact with the respiratory tract, so it encounters antigenic presenting cells, where then they get activate, and then on sensitization they release a bunch of the inflammatory molecule which is known as cytokines, and among them TNF alpha and interleukin 23, they are the major one, which causes the proliferation of NAFT helper cell into its various subsets of helper T cells, such as T helper 1 cells, T helper 2 cells, and T helper 17 cells. And then the asthma follows all these means like the T cell mediated downstream multifactorial inflammatory pathway, which leads to the release of the variety of the inflammatory molecule, which are the cytokines and chemokines, and this all further on progress to the development of asthma. Here is our the structure of the bispec there with a one arm containing anti-interleukin 23 and other arm containing anti-TNF alpha, with the both are linked by the half-life extension partner. So whenever there is a release of interleukin 23 and TNF alpha, our bispec will go and bind to that, and it will stop the further progression of the multifactorial inflammatory pathology of the asthma. So this was our asthma in vivo model. So it was done by the sensitization phase followed by the challenge phase, and then on day 26 the various lung functional parameters were measured, and on day 27 methacholine challenge was done for the confirmation of asthma, and then on the day 28 the animal sacrifice was done by the collection of blood plasma, bulk, and lung tissue for histopathology. So this is a picture of our the bispec means like the each prefill ampule will contains the anti-TNF alpha interleukin 23 bispec depth, and all the study was done in the female vista rats after approval from CCSCA, and the objective of my study was to elucidate the mechanism of bispec in asthma model. So as you know that all the available treatments right from the small molecules to biologics, they are ineffective for treating the asthma as a whole, and they are facing various adverse effects. So coming to the results, so the methacholine challenge was done for the confirmation of asthma. So the PANH value was significantly enhanced in the asthma animal group, and then about the doses of the bispec was able to minimize it. So by the one visual activity, I would like you to show means like what the bispec dab can do for asthma. So at the panel A means like the asthmatic animal reported an increase in exacerbation rate, and in panel B, so means like the bispec dab treated animal greatly have shown the reduction in the exacerbation rate. So the whereas the lung function parameters were measured with the help of the instrument whole body plethysmometer. So the whereas lung function parameters were greatly altered in the case of the disease group, that is asthma group, and then our both the doses of the bispec dab were able to normalize it. Also, the immune cell count were assessed by taking the blood as well as the bulb as the sample. So the asthmatic animal group showed a significantly increase in the number of the immune cell count, where the both the doses of the bispec dab was able to normalize it in the blood as well as in the bulb. So the various biochemical parameters like GSH, SOD, catalase, MDA, nitrate, and MPO were also accessed by taking the lung tissue as a sample. So the asthmatic animal reported significant alteration, while both the doses of our bispec dab was able to normalize it. So the lung histology was done by H&E staining for visualization of the morphology of the lung tissue. So the asthmatic animal group showed an increase in the infiltration of immune cells around the alveolar region, along with the increase in the thickness of the epithelial area of the alveoli. While we can see means like the both the doses of the bispec dab have shown a reduction in immune cell count as well as a reduction in the epithelial surface area. Also, the tolerant blue staining was done for assessment of the mast cell. So the asthmatic animal group showed a significant increase, while both the doses of the bispec dab was able to normalize it, and had caused a reduction in the number of the mast cell. Also, the cytokine analysis of the TNF alpha and interleukin 23, which are both our target of the bispec dab, was done by the ELISA in the plasma, lung tissue, as well as in the bulb. So uh, both the levels were greatly reduced with our bispec dab at both the doses. So means we can show that it has shown. So the future perspective of my study was of my study will be means like those escalation studies needed to be done, safety pharmacology and immunogenicity assessment, validation of our bispec dab of the same in various in vivo asthma models as well as in vitro asthma model. So the discussion is that OLPS induced asthma model was validated, and it was well established, and bispec dab, that is our molecule, that is bispecific antibody. It has tackled the progression of the multifactorial inflammatory pathway of asthma. It has reduced the immune cell count. It has shown the great presence of the lung histology by H&E staining, toluidine blue staining. Also the significant normalization in the various biochemical parameters, as well as through ELISA, the levels of TNF alpha as well as interleukin 23 were greatly reduced. So our study opens a door for the range of opportunities for the treatment of the pulmonary inflammatory disorder by using the various next generation novel biologics as a therapeutic model. And lastly, thank you.
I now invite the judges to ask their question to the respective presenter.
Nice work. I just wanted to ask you a question. How did you administer the drug?
Ma'am, our treatment drug was administered by the subcutaneous route, ma'am, by SpecDep.
Okay, it's a protein, right? That is what I understood out of it.
Yes, ma'am. Yes, ma'am. It is a monoclonal antibody. That is the bispecific antibody.
So, any repercussions of this protein administration, like any allergic reactions or any other thing that you observed in the mice?
Yes, ma'am. Means like the neutrophil and means like the lymphocyte counts have been increased. So it shows that means like some type of immunogenicity is there because this monoclonal antibody is humanized version, ma'am. So if administered in the rat, so it might definitely shows the immunogenicity. And also means like the skin reactions were there severe.
So that did not hamper your observations in any way?
No, ma'am. Means like as for means like the concerning the this means like the biologics. So immunogenicity is the considered to be the normal part because as we can also know means like in the times of the COVID also means like on administration of the vaccine some immune response was shown by us also.
Okay, that's all from me.
Does any other judge want to ask any questions?
नाही मी एवढं मी सिंपलचा करते 0 मध्ये नाही कशी कारण की मागून पण नाही बरोबर
Can you please tell me how did you confirm the induction of asthma in the animal model?
Ma'am, means like by measuring the various lung functional parameter with the help of the instrument whole body plethysmography as well as the methacholine challenge were also done and this is a confirmatory test for the asthma methacholine challenge where means like the pulmonary hyperexacerbation and the pNH is reported in the form of the pNH value. So means like the disease group has shown means like the severe pNH and means like the our treatment group has shown means like the lower the this airway hyperresponsiveness.
Okay. So you have observed all the beneficial effects, but what are the anticipated adverse effects with this?
Ma'am, that means like the specific immunogenicity assessment needs to be done, ma'am. Because means like the concerning with the biologics as a treatment, because of this means like the safety pharmacology as well as the immunogenicity assessment needs to be done, ma'am. That is a future perspective.
On what basis you decided the dose as 2 mg per kg?
Ma'am, means like because means like we have validated by through in vitro screening, ma'am. Because means like at dose of the 1 mg per kg, it was inhibiting the means like the downstream signaling pathway of TNF alpha as well as interleukin 23 also.
Through in vitro?
Yes, ma'am. Yes, through in vitro screening, ma'am.
Hello. Okay.
Thank you, judges, for your valuable question.
Okay,
I would
sir.
like to ask,
Any other question?
Does anyone have any other question?
I would like to call people here. You kindly switch on your camera. We will be presenting your video now.
Greetings to everyone. I'll be delivering the presentation entitled Improved Oral Bioavailability of Venetoclax via Nanostructure Lipid Carriers, In Vitro Lipolysis, Pharmacokinetic and In Situ Single Pass Intestinal Perfusion Studies. So, Venetoclax is a BCS class 4 drug which belongs to very small category of molecules. As is observed in the drug profile, it has low oral bioavailability, approximately 5.4%, and is owing to its poor solubility, poor dissolution, is also a substrate for efflux proteins like AGP, CRP, and it has high first-pass metabolism due to susceptibility to CYP enzymes. Now, to overcome these challenges, we devised a strategy that is the development of venetoclax-loaded nanostructured lipid carriers. The rationale is as follows. When the free drug is administered, because of its low solubility and permeability, is unable to reach the systemic circulation that corresponds to low oral bioavailability. But when such drugs are incorporated into NLCs, now by virtue of several mechanisms, it enhances the lymphatic and ultimately the systemic uptake of these molecules that enhances the overall bioavailability. Now, these mechanisms are as follows. When the drug-loaded NLCs, they come in contact with the GIT environment, they undergo digestion, and the digestive products like monoglycerides, triglycerides, or free fatty acids, they undergo vesicular and micellar formation. Now, the micelles, they enter into the brush border epithelium via unstirred water layer, the phenomena called micellar solubilization. Secondly is the formation of chylomicrons. Along with the drug, phospholipid, and cholesterol, chylomicrons, they are being generated that are readily taken up by the lymphatic vessels, and it ultimately reduces the first pass effect and overall enhancement of bioavailability is achieved. So the objectives of the research are as follows. Firstly, screening of lipids is being done based on the saturation solubility, and it was found that maximum solubility was observed in Econon CC6 in case of the liquid lipids and glycerol monostearate in case of solid ones. Further, the surfactant and co-surfactant, they are being screened based on the particle size and PDI. So basically, blank NLCs, they were prepared and they were then characterized for the particle size and PDI. And among them, vitamin E, TPGS, and transcutol HP, they were found efficient and they were selected for further fabrication. Now, the preparation of VTX-loaded NLCs, they were being done based on hot emulsification homogenization technique. Further, the optimization of venetoclax-loaded NLCs was done firstly on the basis of OVAT approach, where the aqueous phase volume, the sonication time, and the ratio among the surfactant and co-surfactant, they are considered. So it was found that the 20 ml aqueous phase volume with 10 minutes of sonication time and 2:1 ratio of surfactant to co-surfactant, they were efficient and they were further taken for the optimization. So further optimization was done based on DOA approach. The critical material attributes were the one which we have screened in the previous approach. The variables, the independent variables, the dependent variables, and their levels, they are being enlisted in the table given over here. Further, the 3D surface plot shows the particle size, the PDI, and the entrapment efficiency, and it was found that there is a linear correlation in the particle size and PDI, while there is a quadratic relationship in the percentage entrapment. The last figure shows the design space that is being obtained. Further, the model was validated where the predicted values were, in the good agreement with what we had actually observed. The optimized formula for the NLCs were as follows. Further, lyophilization studies were conducted that begin with the screening of protectant, and trehalose to sucrose in 1:1 ratio was selected based on least deviation in PDI and percentage entrapment. This combination was further taken for optimization at different concentration. It was found that 3% ratio of percentage weight by volume of trehalose to sucrose, it had shown least significant deviation in redispersibility index and the entrapment efficiency, and was therefore further taken. Now, moving ahead, the characterization of VTX-loaded NLCs was done. The TEM images confirm the spherical morphology of the nanoparticles, and the DLS analysis has shown it has been done to determine the particle size and the PDI. The FTIR analysis was done, and the disappearance of characteristic peaks of venetoclax indicated that the drug has been encapsulated into the NLCs through the hydrophobic region of the nanoparticles. Following this, the PXRD analysis was done, and the characteristic peak of the excipient, they were absent in the PXRD pattern of NLCs, so which indicates that there is a partial recrystallization of lipids with the, in a less ordered crystals might have occurred. So for the stability analysis in the GI fluids, the formulation is incubated with the SGF and SIF for 2 and 6 hours respectively, and there were very less deviation in the particle size, PDI, and percentage entrapment, and therefore the NLCs were found to be stable. Additionally, the cell culture studies were done, beginning with the cell viability assay. The VTX NLCs, they show higher cell viability in comparison with the free VTX suspension when they both were incubated in the CACO2 cell lines. So following this, the qualitative cell uptake studies were being conducted. And from the overlay plot, it could be evidently it could be observed that there is an efficient internalization of the developed NLC formulation. Furthermore, the quantitative cell uptake studies, support the qualitative uptake study data, wherein the VTX NLC, they had shown nearly 10.62-fold higher uptake in the CACO2 cell lines when compared with the free drugs in 3 hours time span. Further, we move to the in vitro lipolysis studies. Basically, the conventional release and dissolution methods, they are not appropriate for lipid-based formulations. The reason being digestion is the crucial step that determines the in vivo fate of such lipidic formulations. So we have disperse the formulation in a lipolytic media and assess the in vivo fate of such lipidic formulations. Now we can observe that VTX maintain approximately 10.88-fold higher concentration in the aqueous phase as compared to the VTX suspension. And upon lipolysis also, around 64% of VTX was recovered in the aqueous phase, which suggests its accessibility for absorption in the intestine. Moving ahead, the in vivo characterization of the formulation was done. Pharmacokinetic studies were conducted in the SD rats at a dose of 40 milligram per kg. Now it was observed that the formulation was able to achieve nearly 4-folds higher AUC and around 3.8 folds higher Cmax than the free drug suspension. That indicates an increase in the oral bioavailability. Now, additionally, in situ perfusion studies were being conducted. So basically, we passed the formulation and the free drug solution through the intestinal segments. Basically, it was used to determine the permeability and absorption rate constant for the formulation. Now, over here, it was found that nearly around 7.9-fold increase in the permeability and around 2.5-fold increase in the absorption rate constant for the formulation is being observed in comparison to the free drug. Further, the in vivo toxicity studies were done in accordance with the OECD acute toxicity guidelines, and it was observed that all biomarkers were in the normal range, and there were no significant difference that is being observed in case of the VTX NLCs as compared to the free drug. That indicates the its biocompatibility. So to sum up, it was found that the NLCs, they hold a huge potential in delivering such poorly soluble or poorly permeable candidates in a more acceptable manner, as it was observed that in case of venetoclax, there is a significant oral bioavailability enhancement. Additionally, the in vitro lipolysis and in situ perfusion studies, they were also found to be a promising tool for in vivo estimation of such lipidic formulations.
Due to some technical glitch, PG02 is not present in the meeting, and we would like to start with code number PG03 to start their presentation.
Hello.
Hello.
Am I audible? Actually, I'm there in the meeting.
Oh, sorry.
We request you to please rename your site.
I had already renamed it twice, but I don't know what's the technical glitch. It's not updating over here. To me, it is showing that it has the code only.
Okay, then we call upon the judges to for their questions.
Yeah, please.
Any observable adverse effects when you did your animal studies?
No, ma'am. No adverse effects are being observed. All the biomarkers, including the hepatic and as well as the nephrotoxicity markers, they were in quite in the limits only.
Okay. And how did you inject the formulation?
Ma'am, that is through the it is injected orally through oral gavage. It was injected to the animals.
And you've done studies of bioavailability as well, right? You showed your results.
Yes, ma'am. The pharmacokinetic data we have.
Good. Good work.
Thank you.
Any other questions?
So I see presentation because technical issues. Like what studies that
Ma'am, your voice is lagging. Can you please repeat it once?
what are the cell culture studies that you have performed?
Ma'am, that are the CACO2 cell culture studies. We had checked for the cell viability, the uptake studies, qualitative and quantitative both.
Okay, so what was your inference?
Ma'am, in the viability studies, it was evidently observed that the formulation has it has maintained the viability to nearly that of the control, while in case of free drug, there was a decline in the cell viability. Following this, in the uptake studies, in the cell uptake also, we had found in both the quantitative as well as the qualitative as well as quantitative, the formulation is well internalized into the cell, and we had checked it for the its fluorescence as well. So they were, and even the quantitative data supported those findings that the formulation has been internalized. That is what we can observe in the supernatant from the cell lysate.
Okay, so this was again carried out even in the in vivo studies also, no?
Yes, ma'am. That was actually we had done the cell culture, and finally the to address the objective, that is for whether the oral bioavailability advantage is achieved or not, we had carried the in vivo studies.
Okay, okay, thank you.
Oh.
Any more questions? Thank you, judges, for your valuable questions.
Code number PG03, kindly switch on your camera. We will be presenting your video now.
Greetings to all. I'm here to explain about my research topic, exploring the antimicrobial efficacy of Elytraria acaulis and analyzing the phytochemical compounds. So the problem of this research is antibiotic resistance in microorganisms has surged due to the indiscriminate use of antimicrobials and self-medication, which poses a significant global health threat that undermines our ability to treat the common infections. The solution we have given is Elytraria acaulis is a perennial herb which exhibits several medicinal benefits, particularly in managing the diabetes, providing the antioxidant properties, and offering the hepatoprotective effects. Research highlights its potential as a natural remedy, making it a subject of interest in traditional medicine. The urgency for exploring these plant-derived compounds is heightened by the increasing prevalence of multi-drug resistant organisms and the challenge that present in modern medicine. Objectives of this research is to collect the Elytraria acaulis plant and prepare the crude extract using the various solvents such as methanol, acetone, chloroform, and hexane via Sox-slit apparatus to study the antimicrobial activity of plant extract against the urinary tract infection causing bacterial and fungal pathogen. To analyze the phytochemical compound qualitatively and quantitatively in the crude plant extract and isolate the bioactive compounds using techniques such as thin layer chromatography, column chromatography, GCMS, and FTIR. Materials and methods used in this research is what? Healthy leaves of indigenous plant Elytraria acaulis from the Acanthaceae family were collected from Allegheny Hills, and crude extracts were prepared using solvents like hexane, chloroform, acetone, and methanol in Sox-slit apparatus. The antimicrobial activity of these extracts were assessed using the disk diffusion method along with minimum inhibitory concentration test against the selected bacterial and fungal pathogens. Phytochemical analysis was conducted to identify the various compounds present in the extract. Bioactive compounds were isolated and characterized through techniques such as column chromatography, thin layer chromatography, GCMS, and FTIR. The results from this research. This graph exhibits the antibacterial activity of crude extracts of E. acaulis, and it shows that the methanol crude extract exhibits the maximum antibacterial activity against the E. coli with a zone of inhibition of 18 mm, followed by the Klebsiella pneumonia with 14 mm. And this graph shows the minimum inhibitory concentration of E. acaulis against the bacterial pathogen. The MIC values obtained from the present study ranged from 20 to 80 mg per ml against the bacterial pathogen, with the lowest MIC value of 10 mg per ml observed for the methanolic extract against E. coli, P. vulgaris, and S. flexneri. So this graph exhibits the antifungal activity of E. acaulis of crude extracts. At 100 mg concentration, the antifungal activity of crude extract shows maximum zone of inhibition in methanol against the C. albicans of 14 mm zone of inhibition, followed by the C. cruzi and C. tropicalis. And this graph exhibits the minimum inhibitory concentration of E. acaulis against the antifungal pathogen. The MIC of crude extracts ranged between 160 to 20 mg per ml concentration. Least MIC values were observed in the methanol extract of E. acaulis against Aspergillus flavus, Aspergillus fumigatus, Candida albicans, Candida krusei, Candida tropicalis, and Candida glabrata of 10 gram per ml concentration. This table represents the presence of phytochemical compounds in Elytraria acaulis, and it shows the presence of alkaloids, flavonoids, phenols, saponins, steroids, tannins, and carotenoids in Elytraria. This table shows the quantitative phytochemical analysis of E. acaulis. The crude extracts were quantitatively analyzed using various methods, and the total antioxidant activity was maximum 194.58 mg per mg in the E. acaulis extract when compared with the control ascorbic acid. The ascorbic acid value is 125.84 mg per mg. And this picture shows the GCMS analysis of methanol fraction 5 peaks showing the bioactive compounds. To identify the bioactive compounds responsible for the antimicrobial activity, fraction 5 was subjected to GCMS analysis. And the result shows that the compound responsible for antimicrobial activity is 1,2 benzene dicarboxylic acid with a retention time of 24 minutes. This image shows the GCMS analysis of scaling view of 1,2 benzene dicarboxylic acid. This figure shows the peaks for retention time of 32 minutes, and it demonstrates the presence of 1,2 benzene dicarboxylic acid diethyl ester with the molecular formula of C12H14O4 and a molecular weight of 222. This image shows the exterior spectrum of E. acaulis, and we have identified the 6 functional groups from the extract. They are carboxylic acid, alkane and alkyls, ester, amide, alkane and alkenes, and ether. So we conclude that the investigation into the antimicrobial efficacy of Elytraria acaulis reveals the significant potential for this plant as a source of natural antimicrobial agent, particularly against the pathogens associated with the urinary tract infection. The study findings highlights the effectiveness of methanol extract, which exhibited the highest antibacterial and antifungal activities. This conclusion is supported by the presence of bioactive compounds such as alkaloids and phenols, which contributes to the plant's therapeutic properties. Thank you.
But I have a few. Can we start with the questions?
Hi guys, we can start with the question.
Okay. So I have a few questions for you. Did you keep your solvent controls when you did the antimicrobial studies?
Yes, ma'am. We have kept in control.
I think your presentation has out. I cannot see your presentation. So could you help us with where were your solvent controls? in those images?
Positive control we have used as gentamicin for antibacterial activity and for antifungal activity.
Not positive control, solvent control, solvent.
Solvent controls, ma'am. We have used for DMSO, ma'am, for antibacterial activity.
No, but your extracts are made from chloroform, methanol, all these molecules, right? All these solvents, isn't it? Did you remove these solvents completely from your extract?
Yes, ma'am. We have removed it.
Have you checked? Have you
Yes,
checked
ma'am.
for absence of these solvents?
Yes, ma'am. We have checked it.
Okay, because these solvents themselves have some SIDL activity.
Okay, ma'am.
And also you have not presented me table of comparative zones of inhibition of your positive control and your plant extract.
Yes, ma'am.
You've not presented data of zone of inhibition of your positive control versus your plant extract, isn't it?
Irku, ma'am. It is there, ma'am.
Can I see your presentation?
Yes, ma'am. Sure.
Just scroll quickly.
Ma'am, it is there.
Wait. Just stop at any one. Now see here also you've just given, okay, stop here. You said zone of inhibition of 18 plus minus 0.3 followed by Klebsiella. So you've not given how does it compare with the standard positive control.
Ma'am, we have checked it from. Ma'am, there is a table. I mean, this chart we have presented.
Show me the table.
I mean, the graph there is.
Where was the table?
Okay, the positive control is the last column. Wait. Go back. The positive control is always the last column.
Yes, ma'am.
Okay. Also, now, which of these extracts was found to be most potent?
Methanol extract, ma'am.
Methanolic extract was most potent. So, which phytoconstituents did you find in the methanolic extract?
Alkaloids and phenols phytoconstituents, ma'am.
Alkaloids and phenolics. So then how have you reported phthalic acid? If it is alkaloids and phenols, how are you reporting phthalic acid?
Ma'am, I couldn't get your question. Can you come again, please, ma'am?
Towards the end, from your mass analysis, you have reported benzene 1,2 dicarboxylic acid, no?
Yes, ma'am.
So does it fit in as alkaloid or phenolic?
It is both alkaloid and phenolic compounds present in that 1,2 benzene dicarboxylic acid, ma'am.
Are you sure?
Yes, ma'am.
Okay.
You're sure.
Okay. That's all from me.
Okay, ma'am. Thank you, ma'am.
Do any of the other judges have any questions?
So you have mentioned that the methanolic extract is more effective compared to the other fractions, right?
Yes, ma'am.
So do you think that alkaloids and phenols are not present in the other fractions?
It was present, ma'am, but it was in lower amount. So we just suggested that methanol is more present.
Where is that composition? Where is that composition of the percentage of phenols and alkaloids present in different fractions?
It was in next slide, ma'am.
Okay.
It was in slide. It was in slide number 9, ma'am.
Okay.
Bias collected from LX and FTIR.
Okay. So what are the future perspectives of this US study?
And they have studied the antibacterial activity and antifungal activity, antioxidant activity, anti-cancer and anti-inflammatory activities. So in future, we are giving a drug. We will develop a drugs, ma'am, for these act.
Okay. 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 Thank you.
Thank you, ma'am.
Any other questions? Thank you, judges, for your valuable questions.
I will call upon word number PG04 to switch on your camera. We will now be presenting your video.
Hello everyone. The topic of my presentation is the cross-linked hydroxypropyl beta-cyclodextrin nanoparticle for the improved efficacy of venetoclax against the triple negative breast cancer. One of the major type of breast cancer is the triple negative breast cancer, where there is the absence of the molecular targets and the dysregulation of the apoptotic pathway. In the cancer cell, higher concentration of the anti-apoptotic proteins will there, and the less concentration of the pro-apoptotic proteins, which will increases the cell survival and the cell proliferation. The one of the drug from the market is the venetoclax, which is the selective inhibitor of the anti-apoptotic protein, which inhibit the protein-protein interaction between the anti-apoptotic protein and the pro-apoptotic protein. It selectively binds with the BCL2 protein on the surface of the mitochondria and stimulate the apoptotic pathway, which will leads to the cell death. But the problems of the problem associated with the venetoclax venetoclax is the VSS class 4 drug, so it has the low solubility, low permeability, ultimately leads to the low bioavailability. When we give venetoclax in vivo, because of the high log value, it shows the higher metabolism inside the body that can reduces the venetoclax therapeutic efficacy, and venetoclax shows the high adverse effect. To overcome all these challenges, we use the nanoparticulate drug delivery system to deliver the venetoclax at the tumor site. So one of the best approach of the nanoparticle is the beta-cyclodextrin nanoparticles, which has the cage-like structure. Outer surface is the hydrophilic, inner cavity is the hydrophobic. We can load the sufficient high concentration of the venetoclax inside the cavity of the beta-cyclodextrin. So in the formulation we use the hydroxypropyl beta-cyclodextrin as a molecule which has the sufficiently high solubility and high stability than the parent beta-cyclodextrin. We use the cross-linker to convert the cyclodextrin form into the nanocomplex. And this formed nanoparticle, venetoclax-loaded hydroxypropyl beta-cyclodextrin nanoparticle, we administered via IV route, and that can shows the tumor-targeted drug delivery system due to the EPR effect. What is the mechanism of action behind the formation of the nanocomposite? We can keep the basic pH during the formulation that can lead to the release of the proton from the beta-cyclodextrin, and it acts as the nucleophile. And that particular nucleophile molecule acts on the epoxide linkage of the epichlorohydrin, and that can lead to the formation of the nanocomposite. Method used for the preparation of the nanoparticle is the nanoprecipitation method, where we dissolve the TPJs, which acts as the stabilizer, hydroxypropyl beta-cyclodextrin and the cross-linker in the sufficient quantity of water. We can maintain the pH in the basic range to carry out this reaction. We add the drug in the water miscible organic solvent and add into that aqueous phase by using the injection pump. We can keep that reaction for the 12 hours. We kept that reaction for the 12 hours, and finally we add the HCl into it to neutralize the reaction. The optimization parameters are the pH of water, concentration of the EPA, and the weight of the drug. Optimization of this parameter is important to achieve the sufficient size PDI and the entrapment efficiency. Results of optimization: the 20 milligram of the stabilizer, 1:10 ratio of the cyclodextrin and the epichlorohydrin, and the 13 pH were optimized, which achieves the sufficient size and the PDI, which is important for the tumor targeting. optimization of the entrapment efficiency and the optimization of the drug loading. 20% theoretical drug loading gives the sufficient entrapment efficiency, that is almost near to the 80%, and sufficient the practical drug loading, that is almost up to the 16%. Now we evaluate our formulation for the in vitro parameters and the in vivo parameters. First of all, in vitro release study. We carried out in vitro release study by the dialysis bag method with the molecular weight cutoff 12K Dalton by using the phosphate buffer with the sufficient concentration of the twin. The graphical representation shows that we compare the release of the free drug and the nanoparticle. In case of the free drug, rapid release of the drug is seen, and in case of the nanoparticle, initial nanoparticle biphasic release pattern, where biphasic release pattern is there. Initial burst release followed by the sustained release of the drug from the nanoparticle formulation. Further, we carried out the in vivo cell line study in the MDAMB 231 cell line, which is one of the human derived cell line. We carried out the cell-uptake study, qualitative cell-uptake study. In the qualitative cell-uptake study, this pictorial representation shows that the green fluorescence is higher in the venotoclax loaded nanoparticle as compared to the free venotoclax. This means the concentration of the drug accumulated inside the cell is higher in the nanoformulation as compared to the free drug. And in the quantitative cell-uptake study, this graphical representation shows that the concentration of the drug present inside the cell is higher in the case of the nanoparticle as compared to the free drug with respect to the time. Then we move towards the cell cytotoxicity study. This graphical representation shows that more percentage of the viable cells are there with respect to the free venotoclax and as compared to the formulation. And this graph shows that the reduction in the IC50 value for the formulation as compared to the free venotoclax. The IC50 value of a venotoclax was reduced from 49.09 to 9.37 when treated with the nanoparticle as compared to the free drug. Further, we evaluate the apoptosis study by using the annexin V type. This pictorial representation demonstrates that higher apoptotic index is with the nanoformulation as compared with the free venotoclax. Now we move towards the in vivo studies. Initially, we carried out the pharmacokinetic study. Pharmacokinetic study were carried out by using the ESD rats. Sufficient concentration of the sufficient quantity of we can take the sufficient concentration of the sufficient quantity of the blood from the animal with the different time intervals and quantify the concentration of drug within them. We compare the free drug and the free drug treatment and the formulation treatment. The pharmacokinetic parameters were significantly higher in the nanoformulation as compared to the free drug. The pharmacokinetic parameters are the Cmax, AUC, half-life, and the mean resistance time. Now we carried out the pharmacodynamic or anti-tumor efficacy on the BALP-C model, 41 induced BALP-C model. And we evaluate the different pharmacodynamic parameters such as the reduction in the tumor volume, reduction in the tumor burden, and the change in body weight. In all the cases, the formulation shows the higher result as compared to the free venotoclax. Formulation shows the higher reduction in the tumor volume. Formulation shows the higher reduction in the tumor burden. Change in body weight is majorly associated with the free venotoclax. There is a steep increase in the increasing the body weight is there with the venotoclax loaded nanoparticles. Now these two graphs shows that the formulation shows the higher anti-tumor efficacy than the free drug, as the percent tumor growth is higher and the percent tumor inhibition is higher in the nanoparticles, and the tumor weight is very low when we when in the nanoformulation group. Further, we carried out the toxicity studies. For toxicity studies carried out for the hepatotoxicity markers and the nephrotoxicity markers. All the four graphical representation shows that there is a no significant difference between the concentration of the hepatotoxicity markers like the AST, ALT, and nephrotoxicity markers like the blood urea nitrogen and the creatinine when we compare the two groups like the control and the nanoformulation. But the concentration of the all hepatotoxicity and nephrotoxicity marker where is higher in the venetoclax as compared to the formulation. Then we evaluate the as we then we evaluate the inhibitor hemolytic study because our route of administration is the IV. The pictorial representation and the graphical representation shows that there is a significant difference between the percent hemolysis of the PBS and the nanoformulation. I conclude my presentation with the as I develop the venetoclax loaded hydroxypropyl beta cyclohexane nanoparticle, which shows the high drug loading and the tumor target and the tumor targeting. Second one, the formulation showed the biphasic release nature, initial burst release followed by the sustained release. The formulation shows the low toxicity, formulation shows the high pharmacokinetic parameters and the high pharmacodynamic parameters. Thank you.
I now invite the judges to ask their questions to the representative.
Okay, thank you for the presentation. Yeah, you mentioned that cyclodextrin combination for improved efficacy of, well, which is the drug? Well,
Venetoclax.
Venetoclax.
Yes.
So why you have specifically taken up for breast cancer study?
Because this is the triple negative breast cancer, ma'am, and in the triple negative breast cancer, the molecular targets like the estrogen receptor, progesterone receptor, and epithelial growth factor receptors were absent, and it was seen that there was a dysregulation of the anti-apoptotic proteins and the pro-apoptotic proteins. And the venetoclax is the one of the drugs which is selectively binds to the anti-apoptotic protein and which stimulate the activity of the apoptotic protein. So it will stimulate the apoptotic pathway and cell death will be there.
Okay, but that parameters have not been evaluated, no, in the in vivo study.
Because, ma'am, it is supported by literature. The concentration of the anti-apoptotic protein is higher and the concentration of the pro-apoptotic protein is less in the cancer cell, basically in the triple negative breast cancer.
Okay. And while inducing this breast cancer, triple negative 41 cells induced breast cancer,
Yes,
how many days was the induction period?
Induction period is, ma'am, 14 to 15 days, up to which the tumor volume will reach up to 100 millimeter cube. Then we will start our treatment.
What was the cell number?
Cell number is about 10 raised to 5. Yes, up to 10 raised to 5. 1 lakh cell to 3 lakh cells.
So with the 1 lakh cells and 10 days.
15 days, ma'am, almost.
Okay. And you have done one representative, qualitative, and quantitative, no? So there you have just assessed the uptake by the cells,
yes,
right? It
yes,
is not the toxicity. Okay,
no,
in the second part, you have done the cytotoxicity studies, wherein you have not used, apart from this, you have not used any other standard drugs, right?
No, ma'am.
Cytotoxicity studies.
No, ma'am. We only compare the free drug and the drug loaded formulation. We don't use any another drug.
Okay. And on what basis you have decided the dose for the in vivo studies?
The dose is decided according to my human dose. There is the formula for the determining the dose of the animal dose, that is the 5 milligram per kg. The formula is the human dose into the human correction factor divided by the animal correction factor, and correction factor is the correction factor is nothing but the body surface. We can calculate according to the body surface area.
Yeah, fine. But it is a developed new formulation, no?
Yes,
the
ma'am.
cyclodextrin formulation, no? So how
So,
can you correlate?
it's according to ma'am, we according to the pharmacokinetic data, according to the half life and the IC50 value, we determine the dose of our formulation. As if for the free drug we require the 5 milligram per kg, so we can take the for the nanoparticle formulation we can take the equivalent volume which contain the 5 milligram of 5 milligram of drug within the that particular nanoparticles.
Okay, so no acute toxicity studies were conducted.
Yes, ma'am.
Okay, okay, okay. What any have you done any hematological parameters?
Yes, ma'am. Lastly, hemolytic study. We carried out the
What
hemolytic
is
study for the free drug and the
hemolytic?
yes, ma'am.
Okay, not
Because our
hematological parameters.
no, ma'am. We only carried out the hemolytic study to see the effect of the nano formulation on the RBCs
Okay,
because route of
thank you,
administration is IV. Yes. Okay, ma'am.
thank you, thank you.
What about the metabolism? Did you regulate or perform any metabolism studies? Any study on the metabolites? Because you were doing
No,
animal
ma'am.
models as well, no?
Yes, ma'am.
So did you carry out any studies on the metabolism to know how the formulation is being excreted, like whether there are any side effects or any problems?
No, ma'am. We did not carry out any metabolic study that can be.
Okay, thank you.
Thank you, judges, for your valuable questions.
I would now like to invite code number PG05. Kindly switch on your camera. We will be presenting your video now.
Good morning, everyone. Today I am going to present on the topic dual-acting oxaliplatin chlorambucil prodrug-loaded albumin nanoparticles for safer synergistic anti-cancer activity. The developed formulation has been tested in triple-negative breast cancer. The triple-negative breast cancer is sensitive to platinum compounds. That's why oxaliplatin has been chosen. And there are certain challenges with the oxaliplatin. In order to overcome these challenges, it is conjugated with chlorambucil. Both acts on DNA of the tumoral cell, and they are active against TNBC cells. The developed prodrug has shown enhancement in lipophilicity, improvement in half-life, shows synergistic effect, and kinetic inertness has been achieved because of the chlorambucil occupying the axial position of the platinum. And it thus prevents the tumor cell resistance. The albumin nanoparticles has been chosen because it shows dual targeting, both passive as well as active, prevents premature reduction, shows controlled release, it is biodegradable as well as biocompatible. The developed formulation, prodrug-loaded nanoparticle, it enters into the tumor cell via GP60 receptor expressed in endothelial cells. Further, it enters into the cancer cell through receptor-mediated endocytosis. It is released into the tumor cytoplasm and further breaks down into chlorambucil and oxaliplatin in presence of GSH, glutathione and ascorbic acid inside the tumor, and further it damages DNA and causes apoptosis to happen. This is the synthesis pathway of the prodrug. Briefly, oxaliplatin reacts with hydrogen peroxide, forms the weak adduct. This weak adduct is then displaced with chlorambucil and forms the prodrug. This prodrug is characterized using FTR, which shows the presence of additional ester peak, which was absent in chlorambucil and oxaliplatin. Further, there is appearance of ester peak in NMR as well, and the acid peak of chlorambucil has been disappeared. Further, mass spectra has shown the base peak at 1003, which indicates the molecular weight of our prodrug, and the log p value has been enhanced as compared to oxaliplatin. Further, the reduction profile shows that Our prodrug, it breaks in presence of ascorbic acid and GSH, which is present in the tumor cytoplasm. And in comparison to glutathione and ascorbic acid, glutathione shows a rapid reduction rate, thus releases active drugs. Preparation of albumin nanoparticle is through desolvation method, in which desolvating agent is acetone and genipine is used as a cross-linker. This is a TEM image of the formed albumin nanoparticle. These are the critical parameters which are optimized. Firstly, pH. A pH of 9 has shown the optimum size. Polydispersibility index. Good morning, everyone. Dispersibility index, theta potential, entrapment efficiency in practical drug loading. The conformational stability of the protein albumin is checked whether good morning is same is almost same, and the percent alpha helix and the percent beta sheets, that is the second structure of the protein, is in close agreement with the bovine serum albumin as compared to the product loaded nanoparticles, and thus genipine is maintaining the stability. Moving forward to in vitro evaluation, the in vitro release study has shown the sustained release behavior of the product loaded nanoparticles, and it shows release kinetics, which is pepas-salin, indicated by the R-square value of 0.99. The qualitative cell uptake studies has shown that intense green fluorescence is seen in comparison to the pre-treated albumin group, which confirms the cell uptake is via GP60 receptor and spark protein. Further, the prodrug loaded nanoparticles has shown increased cytoplasmic concentration as compared to the prodrug alone in quantitative cell uptake studies, and in cell cytotoxicity studies, the prodrug loaded nanoparticles has shown low IC50 value in comparison to other groups, and very low percent cell viability. Further, the product has shown combination index value less than 1 and dose reduction index value greater than 1, which confirms the achievement of synergistic effect. Further, the apoptosis assay has shown that the prodrug loaded nanoparticles has shown higher apoptosis index as compared to the conjugate in both the cell line 41 and MDA 231 cell line, which are the breast cancer cell line, TNBC cell line. Then, and coming forward to the in vivo studies, in which the anti-cancer efficacy studies has shown that the prodrug loaded nanoparticles, bovine serum albumin nanoparticles, has shown this many times reduction in tumor volume and tumor inhibition rate, lowest tumor burden, as well as there's significant reduction in the tumor weight. And this figure also shows the tumor size is very less in prodrug loaded nanoparticle as compared to the other groups. Further, the toxicity assessment has shown that the prodrug loaded nanoparticles has shown insignificant change in the biomarker levels as compared to the control, and further the organ toxicity has shown same results. There is insignificant change in the microscopic slices. Further, the hemolysis percent hemolysis is in agreement with the control group of phosphate buffer saline, and the morphological assessment also show of RBC also shows that the morphology is not hampered as compared to the other groups. Further, the conclusion is that the product loaded nanoparticles has shown improvement in efficacy, as well as it has shown dual targeting, plus it has also impart safety. but in order to its successful clinical translation, complete toxicity profiling and study of the uptake mechanism as well as multi-dose kinetic studies needs to be done. And further significance of axial ligand by comparison with other platinum prodrugs and other ligands can be done in future. Thank you. These are my additional slides. Thank you.
I will now invite the judges to ask their question to the respective presenter.
Yes, thank you for the presentation. And you have mentioned that your hypothesis, the research hypothesis. So how did you validate your research hypothesis?
As I've said that synergism will be achieved. So for that, the combination index and dose reduction index value has been determined, and the combination index value less than 1, which indicates that the synergism has been achieved, and the combination index value of dose reduction index value of greater than 1 says that there is a reduction in dose. So in this way, synergism has been achieved. Further, the in vitro and in vivo assays has shown that in vitro has shown that there is a uptake via the receptors which takes the albumin, not via the copper transporters, yeah, any other receptors which are getting down regulated in case of resistance. And in vivo studies, there is a reduction in the tumor size. So in this way, it is showing efficacy as well in the TNBC.
Okay. So what was the number of animals used for each group, in each group?
There was 5.
5 in each group.
Yes.
What
Yes.
was the induction rate? Induction rate of the tumor induction rate?
Tumor induction rate.
Yes, among the induced animals, whether it was 100%, 80%, or
Yes, ma'am. Okay. So it was like we have decided it should be around 110 to 118 millimeter cube. So it was 110, so around 90%.
no. My question is no. My question is, so when you inoculated the cells, 41 cells. So what was whether all the animals developed tumors, or out of 5, how many animals developed tumors?
Okay. Yes, ma'am. All have developed tumors. There was no
It was 100%. Induction.
Yes,
Induction was 100%.
yes, ma'am.
Okay.
Because the mice strain which was used is 41. 41 is derived from biopsy, and the biopsy was used as a model. So there was 100% induction rate.
Okay. Thank you. Thank you.
Thank you.
Can you please flash your IR data?
Yes.
Okay, so how did you prove that the ester is formed?
So, ma'am, in chlorambucil, there is the acid peak which is at 1700, and the ester peak which is appearing only in the prodrug, not in the chlorambucil and oxaliplatin additional peak, and this peak is shifted. So because ester has this wave number at 1730, so it has been shifted, which can be seen.
How about the hydroxy peak? Is it there? This is FTIR or affinity?
FTIR, ma'am.
Is FTIR? Because the peaks are
Yes,
not
ma'am.
very sharp. What about the hydroxy peak? In fact, for your chlorambucil also, it's a very, very broad, non-specific kind of. Did you check? Did you do any other check for the ester? Because mass also. Like,
So I did NMR. I did NMR. So
But how do you
and
prove an ester in an NMR?
so ma'am, the appearance of peak at 179, 170, which was early, the earlier it was at 179, and in the prodrug it has shifted to 170, which is showing up
You're
as
talking about C13 or you're talking
yes,
about proton?
13C, 13C, 13,
Much. Okay.
13C.
Okay. Thank you.
Thank you.
Are there any further questions the judges would like to ask?
No, we can proceed.
Thank you, ma'am.
Thank you, judges, for your question.
I now invite Grillo 6 to present.
Greetings to everyone. The topic of my presentation is exploring the potential of gefitinib and betulinic acid co-loaded albumin nanoparticles in triple-negative breast cancer. Triple-negative breast cancer or TNBC is characterized by the absence of HER2, estrogen receptor and progesterone receptor. That is why it is difficult to be treated as there is a limited response to the hormonal therapy. It is an aggressive form of cancer, difficult to diagnose, and the survival chances are also lower as compared to breast cancer, which generates a need to explore therapies for its treatment. Numerous studies have shown overexpression of EGFR. Around 45 to 76% TNBC patients have shown expression of this receptor. So EGFR can be exploited further. The rationale of this study. So gefitinib, which is a small molecule tyrosine kinase inhibitor, can bind to EGFR receptor, but it has to be given in high dose and there is a chance of development of resistance. So it can be given along with betulinic acid, which is a naturally occurring triterpenoid and also has anti-cancer effect of its own, which can help to overcome these problems. But both betulinic acid and gefitinib are highly lipophilic, which exhibits the problem of poor solubility, poor bioavailability, and off-target toxicity. But both gefitinib and betulinic acid binds to albumin, a serum protein, and binding of these drugs will help to overcome this problem. The gefitinib betulinic acid albumin nanoparticles can be generated, which targets the tumor sites by EPR effect. And also, albumin has a self-targeting ability where it targets GP60 and spark protein on the endothelium and the tumor sites respectively. The method of preparation. So the drugs were added to albumin dissolved in NaCl solution, then drop-wise ethanol was added, and then a cross-linker genepin was added, and the formulation was stirred for 24 hours. The following experimental work is done. First, the synergy is determined for gefitinib and betulinic acid, then optimization of nanoparticles, then characterization, followed by storage stability studies, in vitro studies, and in vivo studies. The synergy was determined by cell viability assay, and the CI index values showed strong synergism for the combination of gefitinib and betulinic acid. It was characterized by the size and the PDI of gefitinib betulinic acid albumin nanoparticles or GEFBET ANPs. Then circular dichroism study was performed to check whether there is a change in the structure of albumin after cross-linking with genepin, and it showed almost similar results. Then TEM was performed and the size of the nanoparticles can be seen, and it can also be seen that the size is similar or has optimum PDI. Optimization of GEFBET ANPs were done first for the albumin concentration. Various factors that were considered were the size, PDI, and zeta potential. So 25 mg per ml was the optimum albumin concentration. Then pH 9 was selected as the optimum pH, And additionally, the factors entrapment efficiency and drug loading were also estimated. Then the cross-linker concentration were optimized, and it was the optimum concentration was 150 microgram per ml. The percentage drug loading was optimized, and it was 4% Jef plus 4% betulinic acid. Then the storage stability studies were performed, and to perform the storage stability, first the optimization of the cryoprotectant was done. So trehalose was chosen to be the cryoprotectant. Further, 2.5% concentration of trehalose was selected. Here, the effect of lyophilization on size, PDI, entrapment efficiency, and zeta potential can be seen. So there was no significant change in the sample for the day 0 to that of the end of the 6th month. Then in vitro studies were performed. So release studies was done, and the free drug was released rapidly, whereas the JeffBet ANPs, the release was sustained for about 48 hours in pH 5.5. Further cell uptake studies were performed. So for the qualitative cell uptake studies, the albumin particle showed a higher cellular uptake as compared to the dye. In the quantitative study, the cell uptake for the JeffBet ANPs is significantly higher as compared to the free drug. Then, cell cytotoxicity studies were performed by MTTSA, and hemolytic studies were performed. So, JeffBet ANP showed a higher cytotoxic potential on the cell lines 41 and MDMB 231 cell lines, whereas they showed a lower hemolytic potential on the RBCs. Apoptosis study was performed on both MDA MB231 cell lines and 41 cell lines, and the JeffBet ANP showed higher level of apoptosis as compared to the free drug. Then, for the in vivo studies, pharmacokinetic studies were done, and here it can be seen that the AUC value MRT of the JeffBet ANPs is comparatively higher than to the free drug. Toxicity studies were done. Various biomarkers such as AST, ALT, BUN, and creatinine levels were checked, and it was comparable to the control group for the JeffBet ANPs. Then, in vivo studies, pharmacodynamic studies were performed. The percentage tumor inhibition, tumor volume regression was checked, and here is an image for the same. So the percentage tumor inhibition was significantly higher in the JeffBet ANPs, whereas as compared to the control group or the free drug alone. All those studies were done, and to conclude, JeffBet ANPs were prepared using dissolution method. They were further optimized. They showed high entrapment efficiency, particle size around 165 nanometer with a narrow polydispersibility index and high negative surface charge. Storage stability studies were done. In the in vitro studies, a high anti-cancer activity and lower toxic potential was seen. In the in vivo experiment, a substantial reduction in the tumor volume was seen. That's all from my side. Thank you.
Yes, I now invite the judges to ask
Yes. Yes, thank you. And here you have not mentioned the IC50 value in the in vitro studies, MTTSA, for the both the combination and the free drug.
Hello.
Yes.
Yes, ma'am. I it was also calculated, but I have not mentioned in the presentation.
Okay, what's the difference in the IC50 value?
Ma'am, that IC50 value is the CI is the combination index value that after the synergy, after combination of the drugs,
how much cytotoxic potential they are exhibiting on the cell, and IC50 is the 50% of the uh,
Yes.
IC50. Hello. Am I audible?
No. You are not audible. Yeah, please. Yeah, can you please? Hello? Hello?
Hello.
Hello?
Ma'am,
Yeah.
am I audible?
Yes, now you are audible.
So IC50, the 50% of the cell, the value on which which is lethal to 50% of the cell, or where 50% of the cells are killed.
Yeah. What was the difference? What was the IC50 value for the standard, the free drug and the combination?
Hello.
Yes.
Sorry, ma'am.
Yes,
Can you
please.
repeat? Sorry, ma'am.
What was the IC50 value
That
for
you
the
were
free drug?
asking. It was
Yes.
less than 5.
And the free drug?
The difference.
Yeah. No. What was the approximately? What was the value? Further, how many folds difference was there?
The IC50 value, it was 5 or less than the free drug.
Okay. Okay. So here, how many animals you used for this induction in your study?
5 animals per group.
But for statistical significance, minimum is 6, no?
Yes, ma'am. 6 were asked, but only 5 were approved from the authorities.
Okay. Okay. Thank you. Thank you.
Thank you, ma'am.
Just one question. If you are making albumin nanoparticles, where where all do you think that the nanoparticles can go in the body system?
Ma'am, since the size is around tumor sites have leaked is higher in the nanometer range, is expected to accumulate more in the leaky vasculature side or the tumor side. It will not go in the side.
Is albumin a normal carrier protein?
Sorry, ma'am.
Is albumin a normal carrier protein? Is it distributed throughout the plasma?
Yes, ma'am, it is distributed throughout the plasma.
So then why do you think there would be specifically accumulation in the tumor mass? Why will there not be increased delivery to other organs as well?
Ma'am, because albumin usual size is around 5 to 6 nanometer, and here we have aggregated the albumins and then stabilized by the cross linker. So then the size is increased to 165 nanometer, specifically for the formulation.
Okay.
Thank you, judges, for your valuable questions.
I would now like to call upon code number PG07 to switch on your camera. You will be presenting your video.
Hello everyone. Today I'm going to present my research work titled labeled Free Real-Time Assessment of Tropoid Cell Scintillation. So begin with the placenta. Placenta is the first and largest fetal organ which develops during the pregnancy. Uh, it is made up of multiple villi, starting with the villi core. Villi core is composed of fetal blood vessels and it is lined endothelial cells, which are further lined by the cytotropous cells, which further divides to form the syncytiotrophoblastic multinucleated cell layer, which is directly in contact with the maternal blood. The syncytiotrophoblastic cells performs various crucial functions during the pregnancy, such as endocrine barrier function, nutrient functions, and metabolic functions. Any disruption or defects in the formation of the syncytiotrophoblastic layer may lead to the pregnancy-related harmful outcomes such as implantation failure or IUR, preeclampsia, and preterm births. So, let's understand how these syncytiotrophoblastic cells forms. The mononuclear cytotrophoblast cells come together, fuse to form the multinuclear cell structure known as the syncytiotrophoblastic. To mimic this in vitro condition, there are several cell models available. Among them, Bevo's trophoblast cell line is widely used to study the scintillation in vitro conditions. On treatment with the forskolin, which is adenyl cyclase activator, which tends to Bevo cell fusion and leads to the scintillation. To assess the scintillation changes in vitro condition, there are several methods such as a reduction in E-cadherin, effect in organization. But however, to available conventional method, there are certain limitations as they are qualitative or semi-quantitative measure for the scintillation. Some of them, this measures give just only the number of scintillation fusion events, or these methods majorly all these methods are endpoint assessment. None of them are real-time assessment, giving real-time assessment of scintillation and rate at which scintillation is happening. This leads to the aim of the study to develop method for real-time assessment of true cell scintillation in vitro. So we are going to use the BO cell as a cell model to standardize the scintillation with the forskolin. We perform the immunofluorescence and we check the effect in organization in the BO cells on treatment with forskolin. In control condition, you can observe clearly here the effect in stress fibers are showing at the cell borders to cell borders surrounding individual nuclei, whereas in the control forskolin treated cells, the reduction in the effect in stress fibers was observed and multinucleated cell structure formation was seen. Further, we quantified the fluorescence intensity that shows the reduction in the effect in fluorescence intensity in forskolin treated cells. And then we further uh, we further observe the changes using the mRNA expressions of the scintillation markers. In the first graph here, you can observe the reduction in the junctional protein E-cadherin 1, CDH1 junctional protein, twofold decrease in the forskolin treated cells, whereas in the syncytin 1, the scintillation, another scintillation marker shows the eightfold increase in forskolin treated cells. This conclude that BO cells have successfully differentiated using forskolin. Then we came to our actual aim to assess the changes happening during the scintillation process in the real time. We perform the live cell imaging when we treated with the forskolin. These are the static images of 12-hour interval. Here you can observe in the forskolin treated cells, the cells at the time zero in the both control and forskolin condition, cells are equally dispersed, and in the forskolin treated cells, cells tend to flatten at a faster rate and form the single cell cluster in particular field, whereas in control condition is not happening at that faster rate. Then we question, can we quantify these changes which we are observing through the imaging? Then for the quantification, to answer that quantification question, we use the high content screening system, which comes with the image analysis software. That image analysis software provides us with several parameters to quantify the images, such as cell tracking, granularity, cell area, Then, using that image analysis software, we developed our own customized image analysis pipeline to get scintillizing versus non-scintillizing cells difference in control and force cooling treated cells. Another interesting observation which we observed in the digital phase contrast imaging is the granularity, the change in the granularity in the force cooling treated cell compared to the control. These are the after several permutation and combination, we are able to reach three different parameters that can aid the determination of scintillation. So first is the number of cell cluster available in that same field, and then further, then we used area divided by number of cell cluster as the another parameter, which gives us the significant in difference in scintillizing versus non-scintillizing cells. And the last parameter which we observed in the digital phase contrast imaging is the granularity, which shows significant reduction in the granularity in forcefully treated cells across the time period. Then we question, are these then question come in our mind is, are these changes specific to the scintillization? The pipeline or the analysis which we gives us the results are specific to scintillation. Then we go for the known another inducer for scintillation and another inhibitor for the scintillation. In the inducer one, we are expecting that results will be overlapping with the force cooling, and in the inhibitor one, we are aiming that the analysis pipeline shows us the results overlapping with the control condition, and those changes observed are specific to the force cooling uh, mediated PO cell scintillation are specific to the scintillation, or successfully concluded from these two experiments. Then further, uh, we had question, can we use this system to discover the regulator of scintillation in high throughput manner? So for that, we designed experiment in a such a way that we had four conditions: control, force cooling as a positive control, then a reagent or a compound which we are going to test in alone, and the force cooling plus reagent to test the effect of that reagent on force cooling mediated scintillation. This experiment, for this experiment, I took the compound or reagent which are available for me at that time in the lab. Uh, and we found interesting results. This 14 compounds at in a one experiment shows us three broad categories of the compounds shows showing different effect on the sensitization. We employed the same simple linear regression and calculated the slope to get the rate at which Important process during the pregnancy.
Have you logged in from two devices? I think that is what is creating that echo.
Okay, now, ma'am.
Now you're fine.
PG0 startup, kindly switch on your camera, please.
Please switch on your camera and you can then answer your question. Yes, go on.
So, I have used this. We have developed this analysis pipeline to study scintillation in real time. So, using this method, we can screen the actual process in vitro condition, which happens in vivo condition. So, it is not the cytotoxicity or anything happening. Uh, it's the process which is happening during the.
See, what I understood is you're looking at molecules that can trigger the growth of the sensation, right? That is what your work is about, isn't it?
So basically, we are developing a new approach to study to study the process in vitro condition in the real time. So, if any molecule or any drug that can hamper the that process, we can get the result in the real time because it is continuous and ongoing process in vivo condition. So we can't any drug affect based on the cytotoxicity of the cells because it is the process which is the transition from mononucleated to the multinucleated cells.
But you written pregnancy related complications. So I wanted you to elaborate on that. Like, what is it that you think can be investigated or stopped with your kind of study?
Okay, so if the that process of the transition of the cytotrophoblast to syncytotrophoblast, mononucleated to multinucleated, if any drug we are giving to the pregnant woman, we don't know the effect of that drug on the that transition. So we are developing a new approach to study that affect the drug affect how that drug affect that transition in the real time. Several studies shows that the cytotoxicity of that drug on that cells, but there are less or none studies showing the effect of drugs having on that transition to cytotrophoblast to syncytotrophoblast. So this is the approach we are giving new approach to study the drug screening in the real time.
Okay, thank you.
Yes, it's an interesting work. I just want to know, are there any methods available as of now, or it's a first study in which you are validating this model, in vitro model?
So there are methods, but those methods are endpoint assays. As after 48 hours or after 72 hours, we can study the effect of that drug on scintillation. But this is the first time we are approaching or we are develop the method in the real time. We can assess that changes or the rate at which scintillation got hampered due to the any drug.
Okay, so you have developed it now, but how are you going to validate it? It's going to mimic the actual conditions. How are you going to validate your method?
So we have validated by conventional method the developed analysis which we are showing based on image analysis. So we are validated with previous conventional method. A system is working in our hand, and then we
What was the
use
previous
the known
conventional
inducer
method?
as well as known
No, what was the previous
previous methods are
conventional method? What was the previous, what are
immunofluorescence
the
and
previous,
PCR.
No, could you please tell
Yes,
me
ma'am.
the conventional
Immunofluorescence
methods?
study,
Huh.
immunofluorescence studies, as well as
Okay.
gene expression studies.
Okay.
Those are the
So immuno.
conventional
Huh.
methods to study civilization. But here we are
Okay.
proposing new approach study in real time.
Okay. No, what is that your method? Could you please explain? See, there are different methods, no, by Western blot or gene expression studies, immunofluorescence. So, what is your method of approach?
So here we have developed image analysis method. We by using live cell imaging, we capture the images by interval of 1 hour throughout the process from time zero when we treated the cells with forskolin. From that time point, we capture the every 1 hour interval images up to the 48 hours, and by using those images of the 48 hours in 1 hour interval, we develop image analysis approach. So changes happening throughout the live cell imaging,
Okay.
we capture those changes and develop new approach to study scintillation. There are several parameters by which we calculate, quantify those changes using image analysis.
No, the currently available conventional methods are not analyzing at different time points.
So no, ma'am. Those methods we can apply after several hours only, and those methods gives us just only the fusion events, how much cells get scintillized only. The rate at which scintillation happening, that can't be studied by the conventional methods.
Okay. Okay. And
So
what's the purpose of
as
this? Yeah, please.
from the I give an example. If you do the study by the immunofluorescence, from the immunofluorescence we can get just the fusion events, the number of fusion events from the immunofluorescence. Immunofluorescence
Okay.
can't able to let us know the rate at which scintillation process is happening in the BO cells or the any during the process of scintillation.
Okay, so here it is image analysis, right? So wherein the fusion process and all, but that is okay, fine. Okay, so there also maybe the immunofluorescence Western blot means they'll be assessing the expression of proteins, no? So with that, they'll come to know to what extent it is affecting the process.
Yes, ma'am, that can be done, but this is more uh, tedious or laborious method, and this the method which we have developed. We can use in high throughput screening as well. So at one go, we can screen several drugs in combination as well, and in individual drug also at different concentration as well. Because this is the more of a image analysis, so we have trained the system as in that way. The system gives us the reading in one go for the several drugs.
Okay.
Yes.
So here you are capturing the images and then the images are analyzed using the software, right?
Yes, ma'am. Yes, ma'am. Yes. We are performing live cell imaging for the whole process of scintillation, and then we are aggregating all those images and using the parameter which we found, showing difference in the scintillizing cells versus non-scintillizing cells, and using those parameters we are setting the drug effects.
Okay, so what's next? So now we have done this study and you have proved this is one of the more reliable and more you can assess the exact process. So what is your next step?
So we are going to use this method to the another experiment which is in our lab doing the organ on chip. So we are trying to study this drug screening on the 3D model as well using the develop pipeline, image analysis pipeline. How this method can be.
Okay. Thank you. Thank you.
Welcome.
Thank you, judges, for your valuable question.
I would now like to invite PG08 for their presentation.
Good afternoon, everyone. Today I am here to present the topic of my presentation, exploring treatment potential of docetaxel loaded PEG-PCL nanoparticle for effective management of colorectal cancer. Starting with background and rationale of my research work. In India, the prevalence of colorectal cancer in both sexes is around 4.9%, while in male it was found to be 6.3%. The worldwide prevalence of colorectal cancer is expected to increase by 60% with over 2.2 million new cases by the end of 2030. Currently, there are few marketed formulations for the taxane group of the drug, but most of them fails due to low efficacy and high toxicity. Therefore, the hypothesis of my research work is the preparation of docetaxel PEG-PCL nanoparticles, which will injected via intravenous route. It will get accumulated in the tumor microenvironment. From the tumor microenvironment, it will internalize in the cytoplasm of the colon cell. It will shows the drug release in that. The drug will shows the BCL2 inhibition, increase microtubule stabilization, and disruption of mitochondrial potential, which lead to apoptosis of the cell. Owing to nanometric particle size and encapsulation of drug inside the particle will enhance the anti-cancer efficacy and reduce the toxicity. Then the drug-loaded PEG-PCL nanoparticle prepared by nanoprecipitation method. Various factors involved in the preparation of nanoformulation were optimized using design expert software. The parameter optimized was percent TDL 12%, polymer concentration 8 mg, organic volume 4 ml, aqueous volume 10 ml, infusion rate 1 ml per minute. After the preparation of formulation, the formulation were used for characterization by size and PDI using DLS. The size was found to be 188 nanometer and PDI indicate monodispersity nature of the formulation. The tape image will indicate the spherical particle with uniform size. Following the characterization, live protectant selection will perform based on the reconstitution index and the redispersibility index. In this case, 7.5% sucrose was found to be live protectant of choice, and it will indicate redispersibility index near to 1 and reconstitution index, and it will indicate better reconstitution index. Then the in vitro release will perform at pH 7.4 and 5 at pH 5.5. The in vitro drug release was found to be higher at pH 5.5 compared to pH 7.4. Around 95% of the drug will release at pH 5.5 with the time interval of 192 hours. And the drug release kinetic indicate Korsmeyer-Pipes model for both the pH. Then the in vitro biological characterization was performed on HCT16, ST29 and CT26 cell line. And the percent cell viability was found to be decreased after treatment with DTX PEG-PCL nanoparticle. And the IC50 value for HCT106 is 5 microgram per ml. For ST29 is 6.8 microgram per ml and CT26 6.3 microgram per ml. Then the in vitro cellular uptake study will perform on HCT116, ST29 and CT26 cell line. High cellular uptake was shown after the treatment of coumarin loaded epithelial nanoparticle due to its smaller size and caveolin-mediated endocytosis, and which was found that it will decrease after treatment with genistein. Then the in vitro efficacy will to evaluate in vitro efficacy, apoptosis assay will perform on the same cell line, and it will show the DTX epithelial nanoparticle will show highest apoptotic assay. The high apoptotic assay and the high cellular uptake, it will indicate better in vitro efficacy of the prepared nanoformulation. Then the formulation were analyzed for the hemocompatibility. As shown in the graph, the formulation shows the negligible hemocompatibility after treatment with DTX PCL nanoparticle, while free drug shows a significant hemotoxicity. The morphological evaluation of RBC indicate the DTX PCL nanoparticle shows the hemocompatibility in nature. The in vivo pharmacokinetic study will indicate 3 to 4-fold increase in the AUC, T-half, and MRT of the prepared nanoformulation. The enhanced in vivo pharmacokinetic will lead to enhance the in vivo efficacy of the prepared nanoformulation. The in vivo efficacy will be determined in a 41 induced tumor model. The graph shows the decrease in tumor volume after treatment with DTX PCL nanoparticle. The tumor weight and the tumor burden also significantly decrease after the treatment of DTX PCL nanoparticle. The percent growth inhibition of tumor was also found to be more than 80% for the DTX PCL nanoparticle. To evaluate organ toxicity, ALT, AST, burn, and creatine level was determined over the interval of 7 days, 14 days, and 21 days after the treatment. The level of biomarker in the formulation was found to be increased comparison to the control group. The biomarker level suggests that the organ compatibility for the TTX PCL nanoparticles. It can be concluded as the QBD approach will be better for the optimization and the formulation of hydrophobic drug-loaded nanoparticles. To improve the morphological property and exhibit pH, the nanoparticle indicate improved morphological property and exhibit pH dependent release. The developed formulation possesses superior in vitro and in vivo efficacy with increased pharmacokinetic and safety profile. Thank you. These are some backup slides.
I now invite the judges to ask their questions.
Could you please go on the slide where you showed pH and drug availability, drug release?
Formulation shows the pH dependent release and PCL carrier, which shows the release at pH 5.5. As reported in the literature, the PCL will degraded in acidic environment, and compared to pH 7.4, the pH 5.5 is the tumor microenvironment pH, and the drug will release in that. The most 95% of the drug is released at pH 5.5, which is a
Yeah,
tumor
but-
microenvironment pH.
But you're talking about delivery to the colon, no? So what is the pH in the colon for the molecule, for your formulation to reach the colon?
The pH of colon is 6.8 to 5 or 6.8 to 5, and talking about tumor microenvironment after the cancerous growth, the pH will in the range of 5.5. So the PEGPCL, as reported in the literature, PEGPCL released the drug in drug release at pH 5.5.
Okay, can you go to your in vivo efficacy? The slide of in vivo efficacy.
Next. Next. Next.
So efficacy.
Next to this.
Yeah, here.
Yes, yeah.
So in graph B, you're talking about animal body weight versus time.
Yes, ma'am.
So what is the reason that the animal body weight is increasing in presence of the formulation?
No, ma'am. After the injection of the cell, the animal weight is increases, and we are calculating a tumor burden that which should not be according to according to OECD guidelines. The tumor burden should not be greater than 10%. So that we have the plotted this graph to show that there is not increase in animal weight after injecting cell and after treatment.
Okay, because this weight is giving some other impression. Okay, thank you.
Okay, thank you, ma'am.
Yes, thank you. See, here you have not mentioned the dose at which you have treated the animals.
The dose is 5 mg per kg.
Which one? Free docetaxel?
Yes, for both free docetaxel and according to we have prepared nano formulation of 5 mg per kg.
5 mg per kg. But do you think acute toxicity studies are not required to take up the in vivo studies? Do you think acute toxicity studies are not required to take up the in vivo studies?
Acute toxicity.
Yes.
Acute toxicity is required, ma'am, because the biomarker we have changed. We have checked to is any toxicity of the formulation in the kidney or the liver or not, so that we have to perform a toxicity study that the biomarker, the level of biomarker indicate the formulation will not be toxic for the liver and the kidney.
Okay. Here you have done your, what is your title? Can you please?
Exploring the treatment potential of PEGPCL. Exploring the treatment potential of PEGPCL nanoparticles for the effective management of colorectal cancer.
Yes. Okay. Yes. So you are specific to colorectal cancer, and accordingly you have done in vitro studies on colon cells, colon cancer cells.
Yes.
But what
Yes,
in vivo studies have been done in the breast cancer cells?
No, it's sorry to say, ma'am. It is a CT26 induced cell line. I have by mistakenly take the name 41. It is a CT26 induced cell model.
C18 induced?
Yeah, CT26 induced tumor model for the animal study, not the 41.
Okay, what are the parameters that you have evaluated?
For in vivo efficacy?
Yes, yes, yes.
tumor volume, tumor burden, and inhibition, percent inhibition of the tumor. And the after treatment, the reduction in the tumor also we have determined by the photographs.
Thank you.
Thank you, ma'am.
Any further questions?
No, you can proceed.
Thank you, judges, for your insightful questions. I'd like to note now we'll be taking a 20-minute break. Please make sure that you remain in the meeting and we will resume the presentations after 20 minutes. Thank you.
Judges, with your permission, we would like to resume.
Yeah, we can resume our work.
I would like to invite 4-9 to kind of switch on their video. We will be presenting the
Hello everyone, my code is PG09 and my topic is aggregation induced emission based fluorescence probe for dual sensing of protamine and trypsin. Beginning with rationale, protamine is an arginine-rich pharmaceutical compound used in cardiac surgery, interventional radiology procedures, and antidote for heparin. But it also has undesirable effects such as bradycardia, pulmonary hypertension, and cardiac arrest. Thus, monitoring of protamine is crucial. Secondly, trypsin. Trypsin is a proteolytic enzyme which serves as a significant biomarker in diseases such as pancreatic cancer, cystic fibrosis. Additionally, there is higher level of trypsin in pancreatic disorders. Existing methods for detection of protamine and trypsin face challenges of being expensive and complicated. Thus, there is a need to develop fluorescence-based sensors which have advantages of increased sensitivity, ease of use, low detection level, and operate in biological medium. Aggregation-induced emission. Molecular rotors have efficient non-radiative processes which leads to weak emission intensity in their free state. But when the aggregated molecular level, there is enhancement in the emission intensity, leading to aggregation-induced emission. ALPTP is allyl pyridinium tetraphenyl ethylene. This cationic dye, and DSS dextran sulfate sodium is anionic molecule. These two molecules aggregate and form complex due to oppositely charged and which leads to charge neutralization and increase in the emission intensity. When protamine is added to the system, protamine being cationic will form complex with DSS and break the ALPTP DSS complex. After this, when trypsin is added to the system, trypsin being proteolytic will cleave the bond between protamine and DSS, and ALPTP will again form complex with DSS. Next is results and discussion. In figure 1A, we can see fluorescence emission spectra of ALPTP at varying concentration of DSS, that is 0 to 186.5 nanomolar. Initially, ALPTP has minimal fluorescence intensity, but as the concentration of DSS increases, there is increase in the intensity, which is approximately 15-fold increase. Next is absorbance studies. In figure 1B, absorbance spectra of ALPTP in presence of different concentration of DSS is visible. As the concentration of DSS increases, there is increase in the absorbance at 500 nanometer, which concludes that there is complex formation between ALPTP and DSS. Finally, figure 1C shows time-resolved fluorescence decay trace of ALPTP upon titration with different concentration of DSS. Free ALPTP has fast decay, but as DSS concentration increases, the decay becomes slow, depicted by green, purple, orange, and blue lines. The LOD for this was found to be 0.021 nanomolar. Next is the effect of ionic strength to evaluate the type of interaction between ALPTP and DSS. Increasing concentration of NaCl was added to the system. Figure 1.1a shows fluorescence emission spectra of ALPTP DSS complex at varying concentration of NaCl, that is 0 to 740.74 millimolar. As the concentration of NaCl increases, the fluorescence intensity decreases. Same is observed in absorbance studies. As the NaCl concentration increases, the absorbance at 500 nanometer decreases. Between ALPTP and DSS is electrostatic. As NaCl increases the ionic strength of the solution, And finally, it was confirmed by TCS-PC studies in figure 1.1c. As NaCl concentration increases, the ALP-TP DSS complex breaks, and there is free ALP-TP in the system, which leads to fast decay depicted by red line. Next is to evaluate the stability of the ALP-TP DSS complex. Effect of temperature was monitored. Figure 1.2 shows steady-state fluorescence spectra of ALP-TP DSS complex at various temperature from 25 to 65 degrees Celsius. As the temperature increases, the fluorescence intensity decreases, but the fluorescence intensity does not reach the fluorescent intensity value of free ALP-TP, which concludes that till 65 degrees Celsius, there is no disassembly of the complex, and till 65 degrees Celsius, the complex can be utilized. Next is the effect of pH. Figure 1.3 shows variation intensity at 560 nanometer with variation of pH from 3 to 9. The emission intensity does not show significant changes. Same is observed in figure 1.3b, which shows variation in absorbance at 360 nanometer with variation of pH from 3 to 9. It does not show significant changes in absorbance, which concludes that the ALP-TP DSS complex can be operated from pH 3 to 9. Next is the detection of protamine using ALP-TP DSS complex. Figure 2a shows steady-state fluorescence spectra of ALP-TP DSS complex at varying concentration of protamine, that is 0 to 2.25 micromolar. As the protamine concentration increases, there is decrease in the fluorescence intensity, which concludes that protamine forms complex with DSS and ALP-TP DSS complex breaks, which is again proved by absorbance studies. Figure 2b shows ground state absorbance spectra of ALP-TP DSS complex at varying concentration of protamine. As protamine concentration increases, there is increase in the absorbance at 500 nanometer, which can be due to ALP-TP DSS complex breaking and DSS forming complex with protamine. Finally, it was proved by TCS-PC studies. Figure 2c shows time resolved fluorescence decay trace of ALP-TP DSS complex upon titration with different concentration of protamine. As protamine concentration increases, ALP-TP becomes free, and thus it shows fast decay. Next is the selectivity. Selectivity was evaluated in presence of other proteins such as lysine, arginine, histidine, oval albumin, myoglobin, etc. at concentration of 130.6 micromolar. Figure 2.1 shows that protamine has highest emission intensity as compared to others, which concludes that ALP-TP DSS complex is sensitive for protamine, and which can be due to high positive charge content in protamine as compared to other proteins which have. Next is the detection of trypsin using ALP-TP DSS protamine complex. Figure 3a shows fluorescence emission spectra of ALP-TP DSS protamine complex at varying concentration of trypsin, that is 0 to 450.2 nanomolar. As trypsin concentration increases, fluorescence intensity increases, which proves that trypsin effectively cleaves protamine and DSS bond, and ALP-TP forms complex against with DSS. This was proved by kinetic study at 560 nanometer wavelength for 3000 seconds. As trypsin concentration increases, the emission intensity increases, which is again proved by TCS-PC studies. Figure 3c shows time resolved fluorescence decay trace of ALP-TP DSS system on addition of trypsin at different incubation time. As the time increases, the decay becomes slow, which concludes that ALP-TP forms complex with DSS again. The LOD was found to be 32.19 picomolar. Next is the selectivity. The selectivity was evaluated in presence of other enzymes such as acid phosphatase, lipase, pepsin, alpha amylase at concentration of 612.8 nanomolar. Figure 3.1 shows that trypsin has highest emission intensity as compared to others, which proves that the ALP-TP DSS protamine complex is selective for trypsin. Next is the inhibition assay. In order to evaluate the inhibition action of benzimidine, which is a known inhibitor of trypsin, figure 3.2a shows inhibition of trypsin activity measured as fluorescence decay at various concentration of benzimidine. As the concentration of benzimidine increases from 0 to 0.87 millimolar, the fluorescence intensity decreases, which proves that benzimidine inhibits trypsin, and trypsin does not perform its role of cleaving protamine. Figure 3.2b shows the response plot illustrating the percentage inhibition. As the concentration of benzimidine increases, percentage inhibition also increases. Finally, TCSPC studies reveal that in absence of benzimidine, there is slow decay, that is ALTP and TSS forms complex again. But as the benzimidine is added to the system, it inhibits trypsin, and trypsin does not cleave protamine, and ALPTP is free. Thus, there is fast decay. Next is detection of trypsin in real matrix. Figure 4a shows activity of trypsin measured as fluorescence decay in 10% human urine matrix of ALPTP DSS protamine complex at varying concentration of trypsin. As the concentration of trypsin increases, the fluorescence intensity increases. This is the linear plot of intensity versus trypsin concentration. The LOD was found to be 29.3 picomolar. This is the working of the sensor. ALPTP DSS forms aggregation-induced emission, thus there is fluorescence on. When protamine is added to this system, there is fluorescence off because protamine forms complex with DSS. Again, then trypsin is added, there is again fluorescence on because ALTP again forms complex with DSS. This could be the future scope of this sensor. Step 1 is to take patient urine sample. Step 2 is to add sensor, and step 3 is to take reading. Conclusion, novel sensor for sensitive and selective detection of protamine based on ALPTP DSS employing aggregation-induced emission was developed. The sensor had good stability and selectivity. The sensor successfully detected trypsin in human urine samples. The sensor is a simple, cost-effective, and sensitive alternative to traditional methods. These are my references. Thank you.
I would like to invite the judges to ask the question.
So I think you showed that it is stable over a wide range of pH, right?
Yes, ma'am.
So in that case, buffer is not an important component of your experimental protocol.
Yes, tris buffer was used at pH 8.2.
So why did you have the need to use tris buffer? If your graph showed almost no effect of pH, right?
Yes.
So what happened when you performed the same experiment in absence of tris buffer?
So actually, the system is quite stable from pH 3 to 9, but still it was performed in tris buffer only.
And how about trypsin from urine samples? You said you also could analyze trypsin from urine. The pH of urine is acidic, right? So then what about tris over there?
10% human urine was used, so it was diluted in tris buffer urine. 10%.
So artificial urine samples you used?
No, like urine was diluted. Yes. Actual urine was diluted in tris buffer.
Okay. Okay, thank you.
Yes, could you please tell me the significance of the study once again?
Can you repeat your question?
The significance of your study, could you please elaborate?
Yes, so protamine is an arginine-rich pharmaceutical compound. It is used in cardiac surgery and also an antidote for heparin. But it has undesirable side effects such as bradycardia, pulmonary hypertension. Therefore, monitoring of protamine is crucial. Secondly, trypsin is a proteolytic enzyme which is a significant biomarker for pancreatic cancer, cystic fibrosis, etc. And additionally, in pancreatic disorders, there is higher level of trypsin in urine. And the conventional methods such as ELISA, capillary electrophoresis have disadvantages of being expensive and complicated. That's why fluorescence-based sensor was developed for detection of protamine and trypsin.
Okay, now what are the currently available methods for detection of these two in the biological fluids?
Commercial kits are available for detection of protamine and trypsin from Sigma Eldridge and Thermo Fisher. Uh, they are also based on, I guess, Thermo Fisher one is based on fluorescence, but our method has less LOD value, thus it is more sensitive.
Pardon? Your method is more sensitive than the currently available method.
Yes, the LOD is less, that's why.
LOD is? LOD means?
Less. Limit of detection.
Okay. Okay. Means this is very sensitive compared to the currently available.
Yes, and there are also other research on fluorescence-based sensor for detection of trypsin, but they face the challenges of like they cannot be applied in real sample analysis because of complex media like urine, etc. But our sensor can be utilized in urine also. It can detect trypsin.
Okay. Now, did you compare both, whatever your method and the commercially available method, currently available method?
No, comparison study was not done. Only literature survey was done.
Yes. Okay. Thank you.
Is this method also useful in detecting the enzymes from biological fluids?
No, it is used for detection of trypsin in urine because in pancreatic disorders there is higher levels of trypsin in urine.
Okay, but not from biological samples, not from plasma.
Plasma study was not done, only urine was done.
Okay. Okay, thank you.
Thank you, judges, for your valuable question. Code number PG10, kindly switch on your camera. We will be presenting your video now.
Hello, today I'll be discussing on synthesis, characterization, and targeted drug delivery of doxorubicin hydrochloride loaded down convergent and up convergent nanoparticles. These are the contents that I'll be discussing today. First, let's look into what are upconvergent and downconvergent nanoparticles. Upconvergent nanoparticles means when and lower energy is supplied and it emits higher energy, whereas downconversion means when a higher energy is supplied and lower energy is emitted. The aim of my project is to prepare and characterize downconvergent and upconvergent nanoparticles and their application in targeting, imaging, and cancer management. The objective of my project is to first prepare and optimize the upconvergent and downconvergent nanoparticles, then to characterize upconvergent and downconvergent nanoparticles loaded with anti-cancer drug, and then to evaluate the efficacy of nanoparticles and their application in targeting, imaging, and cancer management. Let's first look into the rationale of the project. So first of all,
Dr. Rajkumar, are you would like to ask a question? You can go ahead for PG9. We aren't able to hear you. May you please rejoin the meeting?
Hello, can you repeat? Can you repeat?
May you please unmute your mic?
Hello.
We are able to hear you. May you leave the meeting and rejoin?
Is that topic? So can you please put the questions in the chat?
We'll proceed with PG 10, and anyone can ask any question after the presentation.
Hello, today I'll be discussing on synthesis, characterization, and targeted drug delivery of doxorubicin hydrochloride loaded down convergent and up convergent nanoparticles. These are the contents that I'll be discussing today. First, let's look into what are upconvergent and downconvergent nanoparticles. Upconvergent nanoparticles means when a lower energy is supplied and it emits higher energy, whereas downconvergent means when a higher energy is supplied and lower energy is emitted. The aim of my project is to prepare and characterize downconvergent and upconvergent nanoparticles and their application in targeting, imaging, and cancer management. The objective of my project is to first prepare and optimize the upconvergent and downconvergent nanoparticles, then to characterize upconvergent and downconvergent nanoparticles loaded with anti-cancer drug, and then to evaluate the efficacy of nanoparticles and their application in targeting, imaging, and cancer management. Let's first look into the rationale of the project. So first of all, this project deals with targeted drug delivery using folic acid conjugation. Secondly, it also deals with cell imaging due to the emission of the spectra in a visible spectra range. And lastly, photothermal therapy by caused due to downconvergent nanoparticles. When 980 nanometer is supplied to the upconvergent, downconvergent nanoparticles, it emits green luminescence, which helps in imaging, and also emits heat, which helps in photothermal therapy. The research design and methodology of this project is first creation of synthesis of downconvergent nanoparticles. Firstly, lanthanide precursors with oleic acid and 1-octadecene is used. When it is heated to 120 degrees Celsius for 1 hour, and then methanol containing NaOH and NH4F is added, and then it is heated to 80 to 100 degrees Celsius for 1 hour for the evaporation of methanol. Then further, it is heated at 300 degrees Celsius for 1 hour, then cooled and washed with ethanol and centrifuged. This leads to the formation of down-convergent nanoparticles. For the synthesis of core shell nanoparticles, that is down-conversion loaded with up-conversion, we take lanthanide precursors, again using oleic acid as a capping agent and 1-octadecene as a solvent. Then we heat it at 120 degrees Celsius for 1 hour. Then we add methanol with NaOH and NH4F, which helps in redox reaction. Then we again heat it at 80 to 100 degrees Celsius for 1 hour, which helps in evaporation of methanol. Then we add down conversion nanoparticles and then we heat it for 1 hour at 120 degrees Celsius. Then we heat it at 300 degrees Celsius for 1 hour and then we cool the mixture and then we wash it with ethanol and centrifuge, which leads to the formation of the core shell nanoparticles. Then we coat it with silica to increase the hydrophilicity of the nanoparticles, as the up-convergent down-convergent nanoparticles are hydrophobic in nature. We use CTAP for removing the oleic acid out of it and then adding the silica coating on it. Ammonia is used as a basic catalyst and then TUS is used for silica coating. And then we heat it for 500 degrees Celsius to evaporation of all the CTAP. For amine coating, we use APTMS, which is 3-aminopropyl trimethoxysilane, which we take nanoparticles and water and we stir it for 24 hours. Then we add APTMS and stir it again for 24 hours, which leads to nanoparticles coated with silica and amine. Amine coating is generally used for proper conjugation of folic acid to the nanoparticles. Folic acid, which is generally used for target delivery. We first take folic acid and water and stir it. Then we add EDC and NHS, which helps in coupling the molecule, the folic acid molecule to the nanoparticles. It acts as a coupling agent. Then we stir it for 2 hours. Then we add 30 milligrams of nanoparticles. And then we stir it for 24 hours, which gives us folic acid conjugated nanoparticles. This entire reaction is carried out in dark condition because folic acid is photolabile. Next, drug loading. We have our choice of drug in this experiment is doxorubicin hydrochloride. 10 ml of folic acid coated nanoparticles are used. Then it is dissolved or dispersed in 3 ml of water and then it's stirred for 2 hours. Then we slowly add doxorubicin into it and stir it for 24 hours. Below are the pictures of first nanoparticles dissolved in folic acid, then after the addition of doxorubicin, and then finally the powder, which contains nanoparticles coated with mesoporous silica, amine, folic acid, and then loaded with doxorubicin. The tests conducted are first photoluminescence. As you can see in the photoluminescence spectrum, 4 peaks are observed, out of which peak C, which emits green emission at 545 nanometers, is very much prominent. That's why the particle shows higher green luminescence, which helps in bioimaging. In FTIR, first UCNP coated with DCNP coated with mesoporous silica are checked, in which we can see SiOH peak at the desirable wavelength. Then mesoporous silica and amine coating was also checked, in which we can see amine bend, and then UCNP coated with amine and silica plus folic acid, we see desirable CC aromatic stretch. Zeta potential was done. The main reason for conducting zeta potential was to check the affinity of the various coating to the nanoparticles. And first, we can see nanoparticles coated with mesoporous silica shows minus 27.8. Then silica coating plus amine coating shows 15.7, which shows the close affinity of amine to silica. And then silica, amine, folic acid coating shows 3.45, which again shows that the folic acid and doxorubicin drug is also completely coated to the nanoparticles. In TEM images, we see the 10 images of core shell around 20 to 25 nanometers, and then with mesoporous coating, the nanometer is changed to 85 nanometers. The size of the particles are changed to 85 nanometers. In MRI images, we see that 2 millimole shows most prominent NMR spectra, and this spectra was calculated at different concentration, first 1 millimolar, 2 millimolar, and 0.5 millimolar, out of which 2 millimolar shows prominent MRI spectra. In XRD, we see the graph, which shows us that it gives beta hexagonal phase of NAGDF4, which is our core, which is reported in PDFs 270699. It shows beta hexagonal phase, which is most desirable and stable structure. Drug loading was found to be 25.91% and entrapment efficiency was found to be 99.85%. In vitro release was conducted, in which it was found that it follows Prosmapepal's release, was the most desirable release. And it was conducted into two media, that is buffer of 7.4 and 5.5. 7.4 is physiological buffer range and 5.5 is the cancer microenvironment range. In sterility testing, first the nanoparticles were UV sterilized for 30 minutes and then they were tested for Bacillus subtilis and for fungi Candida albicans in different medias. And after 14 days, the evaluation was done for 14 days and it was seen that no bacterial fungal growth was observed in the test formulation. In headchem studies, it was conducted to check the irritancy of the formulation, in which we see the nanoparticles and API itself are not irritant. Thus, I would like to conclude this presentation by stating that the upconversion and downconversion nanoparticles were synthesized using thermolysis method, and the various tests was done like luminescence, particle size, XRD to check its proper behavior. Then the hydrophobic shell was further coated with silica coating to achieve hydrophilicity, and then targeting was done using folate receptors. Gadolinium was used as an imaging agent, and then doxorubicin was used as a drug. In vitro release was around 94.58% in the end of 48 hours at pH 5.5, which is an optimum pH for the cancer cells. And thus, it is shown that this upconversion, downconversion core shell loaded with doxorubicin is best suited for the management of cancer as a nanotheranostic agent. These are the references of the presentation, few references for the presentation. Thank you.
I now invite the judges to ask their questions to the respective presenters.
Which type of the nanoparticle is useful for management of cancer? Upconversion or downconversion?
So, ma'am, in this we have used both upconversion and downconversion nanoparticles. So, upconversion is basically for imaging purpose, and downconversion nanoparticle is for photothermal therapy.
And what is the life of this luminescence?
Ma'am, actually, we haven't checked yet the half-life of the luminescence, but it was found via the luminescence study that we have done that it was comparatively to the rest of the diet, it is more stable.
Can you come again with that? I think there's some background noise.
Like I mentioned, that the dyes that are used in commercially, this is a better and more photostable compared to the commercially available dyes.
What is the photosensitivity of doxorubicin?
So now actually doxorubicin gets degraded in the presence of external source of light. Therefore, the reaction was carried out in dark conditions.
So when it is in combination with this nanoparticle, what is the stability of doxorubicin inside this nanoparticle?
So ma'am, actually in I'll just want to get my videos on. Hello. Now is it visible?
Yes, it is.
So basically, when the nanoparticles are combined with the doxorubicin, we had done in vitro studies for 48 hours, and the drug was stable and it showed the desirable peaks.
But have you done stability studies on the formulation?
No, we have not done the stability studies.
Okay, thank you.
Yes, it's interesting. Yeah, I think I request the organizers to mute Hello? Yeah, yeah. Thank you. Yeah, you have mentioned that both upconversion and conversion is for the targeted therapy, or it's only down conversion is for the targeted treatment approach.
So, ma'am,
Am I
basically,
audible?
both the nanoparticles. Yes, ma'am, you're audible. Basically, on
Yeah.
down conversion nanoparticles, up conversion, on down conversion, stock to the up conversion is coated, and the target delivery is achieved due to folic acid conjugation.
Could you please repeat? Down conversion is
The up conversion nanoparticles are coated on the down conversion nanoparticles, and the targeted delivery is achieved due to folic acid conjugation.
what's the folic acid conjugates? And you
Yes,
have
folic acid
mentioned in the you have mentioned in the conclusion part also folate receptors. So what is that? Are you deviating from that because the mechanism is what you are using doxorubicin and you are targeting folate receptor.
Basically, in most of the cancer, especially breast cancer, it is seen that an overexpression of folate receptors is observed. Because of that reason, we have conjugated it with folic acid. And due to that mechanism,
Okay.
the target delivery is achieved.
Okay. No, what is the current literature reveals about this upregulation and downregulation, where the doxorubicin has already been, no, the nanoformulation of this downregulation nanoparticles has already been prepared. What's the literature about this concept?
Basically, this nanoparticles have also been studied for cell line studies and for in vitro studies, in vitro and in vivo studies for animals, and it has shown significantly better compared to the normal, like the commercially available dyes in the comparisons like parameters like biocompatibility and the stability or the photostability of the nanoparticles in the animal models.
So what are the approach?
Sorry, ma'am, could you please repeat the question? I couldn't hear.
Limit with this treat approach or whatever this concept, this down regulation or up regulation nanoparticles.
Ma'am, sorry, can you please again repeat the question? Your voice was cracking.
No, see, you have mentioned about the advantages of this up regulation and down regulation. Am I audible? Hello.
Yes, ma'am. Now you're audible.
Am I audible?
Yes, ma'am.
Yeah. So you are mentioning about the advantages of this up regulation and down regulation concept, right? So my question is, so what are the limitations of this according to you, based on the literature? Limitations.
So,
are there any demerits?
One of the ma'am, one of the limitations that I think still needs to be worked on is the method, which is it's a little bit lengthy to get it into market and for industrial application, and even the cost. Basically, the lanthanide series is being used for this, so the cost might also be one of the limitations. So we even need to work on that.
Okay. Thank you. Thank you.
Thank you.
Do the judges have any other question?
I think sir is, you can ask sir.
Sir, are you able to speak? Are you able to unmute your mic? So are you able to speak or should we proceed with the next presenter? May you please text in the chat? Thank you, judges, for your valuable question.
Please note, PG11 is unfortunately not available. So now we're moving forward to PG12. We kindly request you to switch on your camera. We will be presenting your video now.
Hello everyone. My research topic is intranasal delivery of curcumin-loaded functionalized multi-walled carbon nanotube for Alzheimer's disease management. Beginning with background and rationale, curcumin has this inherent effect and inherent activity of tau inhibition, and it has antioxidant properties. It has therapeutic effects and shows anti-inflammatory activities in the body, and it is also proven to show amyloid beta inhibition, which is helpful to elevate the Alzheimer's disease and its management. Also, it is an acetylcholinesterase inhibition and modulates the microglia. But it has some drawbacks such as poor solubility and poor systemic bioavailability and poor permeability. So to tackle this problem, multi-walled carbon nanotubes comes into the picture, as multi-walled carbon nanotubes has a high surface area, and it is also biocompatible and directly interacts with the amyloid beta plaque, which helps in the management of the Alzheimer's disease. This multi-walled carbon nanotubes has high drug loading capacity, and the drug delivery is target specific and helps in the loading of the curcumin and deliver it to the targeted site. Intranasal route of administration has several advantages over conventional dosage forms because it is non-invasive. It passes the first pass metabolism, and also bypassing the blood-brain barrier helps the curcumin to reach the targeted site, and has fewer side effects because it makes sure that the curcumin or other drug which is loaded in the multi-walled carbon nanotubes and delivered through intranasal delivery only it reaches into the target specific site, and it does not reach into the other periphery of the system, which causes less side effects. Moving on to the next part, that is methodology. That consists of functionalization of the multi-walled carbon nanotube initially. The acid functionalization of the multi-walled carbon nanotube is necessary so as to get attached with the drug, and for this polyethylene glycol comes into the picture, because polyethylene glycol makes the multi-walled carbon nanotubes less toxic, more dispersible in the biological environment, and helps to ensure the curcumin is attached, because functionalization of the multi-walled carbon nanotubes introduces the hydroxyl groups and makes it available for the drug to get loaded. And polyethylene glycol on the other side helps the mucus permeability in the nasal tract, and also reduce the toxicity of the multi-walled carbon nanotube. Here, polyethylene glycol is of grade 600. So, multi-walled carbon nanotube is acid functionalized with the help of sulfuric acid and nitric acid in 3:1 ratio. It is subjected to sonication for 7 hours, and further, it is subjected to the centrifugation, in which it is neutralized using NOH, and further washed off with the water so as to remove the residues, if any. Further, this multi-walled carbon nanotube, which is functionalized, it is then mixed with the polyethylene glycol of the adequate quantity, and then pegylation is done of this functionalized multi-walled carbon nanotubes to get ready with for the loading of the curcumin in it. The trial batches of the curcumin loaded multi-walled carbon nanotube consisted of 2 mg of curcumin, in which 5 ml of ethanol was used to get solubilize the curcumin, and then 2.2 mg of multi-walled carbon nanotube, which is pegylated, is subjected to sonication for 2 hours, and further it is subjected to microfluidization in order to reduce the particle size and for the uniform mixture and dispersion of the formulation. This microfluidization is used at 20,000 psi for 8 cycles. This is the FTIR of the formulation and the polyethylene glycol, curcumin and multi-walled carbon nanotube in one spectrum. It is overlaid spectra. So in this we can see that multi-walled carbon nanotube does not exhibit any characteristic peaks, whereas the curcumin has some characteristic peaks because of its carboxylic and hydroxyl groups attached and aromatic rings near 2500 and 3000. Further, we can see that the formulation, that is multi-walled carbon nanotube, and attached with the PEG and the drug loaded in it, we can see the peaks of curcumin is not that specific because it gets entrapped into the multi-walled carbon nanotube. And yeah. Further, in results and discussion, we have done solubility of curcumin because it is soluble in ethanol and not soluble in other aqueous phases. DSC curve of the curcumin depicts the thermal degradation point at around peak temperature 184 degrees Celsius. The FTIR spectrum of curcumin shows that it is of purity of the curcumin. That is, it ensures that around 3000 we can see the carboxylic peaks are there, 3500, 3000, and near 1600 we can see the aromatic groups are attached. The another spectra depicts the multi-walled carbon nanotube, which is treated with the acid, that is H2SO4 and nitric acid, 3:1 ratio. Evaluation of the trial batches. The trial batches which were evaluated, in which entrapment efficiency is around 78% and the desired zeta potential was around -32 millivolt, and the particle size was around 100 nanometer. Optimization of the formulation was further done with the design of experiments using the Design Expert software version 13, and 3:2 factorial design was applied, in which these independent variables were varied across 3 levels. Independent variables here are curcumin and multi-walled carbon nanotubes, which are functionalized and pegylated. And dependent variables are 3, that is particle size, zeta potential, and entrapment efficiency. These are the optimized results of the formulation by DOE, in which further, depending on the desirability, the optimized batch is chosen. And here the implication of variations in particle size, zeta potential, and entrapment efficiency, we can see there is changes in these 3 parameters. And further, according to the desired and required particle size and zeta potential and entrapment efficiency of the drug, these are the predicted and the experimental values, and this is the optimized batch. Further, the transmission and electron microscopy of the formulation was done to ensure the internal structure of the carbon nanotubes and the curcumin which is loaded in it. We can see the dark parts that is there. Further, to evaluate the antioxidant activity of the curcumin, which is loaded in the multi-walled carbon nanotube in the formulation, it is evaluated using the ascorbic acid as a standard. And by DPPH assay, we can see around more than 60%, we can see the antioxidant scavenging activity of the curcumin. Further, in vitro drug release was done using dialysis bag and in France diffusion cell. So it was done in two different buffers, that is at pH 5.5 and 7.4. We can see the percentage cumulative release of the drug for the pH 5.5 was 95%, and for the 7.4 pH it was 89.9% respectively. Further, for the release kinetics, it follows Korsmeyer-Peppas model of the drug release, because the model is used to describe the diffusion of the drug from the carbon nanotube, and that shows that diffusion is dissolution controlled release, hence it follows the Korsmeyer-Peppas model. Ex vivo permeability study was performed using goat's nasal mucosa, and the percentage cumulative release was around 80%. In vitro cell line studies were done using PC12 cells, that is pheochromocytoma rat cells, in which cell viability and neuroprotective study was done on these cells. The differentiated cells were taken and they were subjected to different concentrations of the curcumin as seen here. And these bright green images are of the viable cells, and the dark red images are the cells that are destroyed and that are subjected to degradation. The neuroprotective study was done using H2O2 induced PC12 cell, and IC50 was calculated. Pharmacokinetic parameters. In pharmacokinetic parameters, the male vista rats were taken, and from there the blood was withdrawn from the retroorbital plexus root, and it was further the plasma. Before that, the rats were administered with the curcumin solution and the formulation, and both were evaluated at the different time points. And we can see that the plasma concentration versus time profile of the group 2 curcumin solution and group 3 optimized batch of the curcumin solution is depicted here. Further, brain distribution study was done. Here actually 3 groups were taken. Group 1 was control, group 2 they were administered with the curcumin solution 10 mg per kg, and in the third group the formulation was administered, and the brain was removed and it was homogenized using the saline solution. Further, it was homogenized, and the plasma, the supernatant, was taken and it was evaluated using the HPLC column C18, and the mobile phase was acetonitrile and OPA water in 60-40 ratio. We can see here curcumin solution in the brain, and the formulation in the brain. The formulation in the brain exhibited a higher distribution in the brain than that of the curcumin solution, and hence this PEG significantly increased the brain uptake of the curcumin that is loaded in the pegylated multi-walled carbon nanotubes. Further, the stability studies were done according to ICH Q&A guidelines, and in this 3 different temperatures the formulation was stored, and we can see that during the observation there was no agglomeration of the multi-walled carbon tubes in the formulation, and the particles at zeta potential and the drug content was also evaluated on these 3 different months. Conclusion, we can conclude by saying that curcumin loaded multi-walled carbon nanotubes which were pegylated were successfully developed, and along with that cell viability studies, neuroprotective studies backed that the cells are more viable in this, and there are not the toxic effect of the carbon nanotubes is here depressed because of the introduction of the polyethylene glycol, and further the batch was optimized using design expert software. And overall, the T-half also was found to be higher than the optimized batch of the curcumin loaded multi-walled carbon nanotube as compared to the curcumin solution. And further, the stability studies were done of the formulation according to the guidelines. Thank you.
We request the judges for the questions.
Yes, thank you. Your presentation is your research work. You mentioned it is against Alzheimer's disease, no? What's your title?
Your mic is on mute. Please unmute yourself.
Am I audible?
Yes, ma'am. It is. Yeah. Yes, ma'am. You are audible. It is for Alzheimer's disease management.
Yeah, but you have not evaluated the same in the disease model, no? Animal model, no?
Yes, ma'am. The disease was not induced actually.
So, efficacy means it is the exposure. You have done animal brain distribution studies, right? So,
Yes,
how did
ma'am.
you? What was the route? Means the exposure, mode of exposure, and what was the amount, the amount of that combination that you administered.
So, ma'am, the drug was administered intranasally. 10 mg per kg was taken.
How did you administer?
Intranasally, ma'am, with the help of intranasally it was administered. Yeah.
Pipette, using pipettes.
Yeah. It was another type of pipette. Actually.
Okay.
I could not recall
amount of drug administered.
10
what was the
mg.
amount that you? No, 10 mg
per
is
kg.
fine. No, it's in the solution. No, it was a solution form. No.
Yes, ma'am. This dispersion form, sort of dispersion.
No, the volume.
Liquid.
The liquid. No, how much volume was administered?
10 microliter.
10 microliter consisting of 10 mg.
Yes.
Okay.
Does curcumin have efficacy at that concentration?
Yes, it had shown activities at that concentration.
Otherwise, does curcumin have can it cross the blood-brain barrier? Only curcumin. Can it cross the BBB?
So curcumin crossed the BBB with like when it is administered along with the multi-walled carbon nanotubes, the brain distribution was there as when it was compared to curcumin solution, which was made different, and another was there, which was the formulation. So these both were compared, and brain distribution of the formulation was fourfold more than that of the only curcumin.
Yeah, so does plain curcumin cross the blood-brain barrier?
Not as comparatively to multi-walled with the multi-walled carbon nanotube. If single, actually the bioavailability of this curcumin in the brain is not that much as comparatively to the formula.
What is the fate of the carbon?
Cannot pass the blood-brain barrier because it gets eliminated from the body.
what is the fate of the carbon nanotube after it enters the brain?
Wait,
Fate.
Fate.
Metabolic
Okay.
fate.
After it enters the brain, it can be engulfed by microglia cells, which are immune cells present in the brain, and other immune cells. Phagocytosis happens to this. From the literature review, I have concluded that it can be eliminated. Also, the raw multi-wall carbon nanotube has toxicity to certain level. To reduce that toxicity, polyethylene glycol was used.
Okay, thank you.
Thank you, judges, for your valuable questions. I would now like to invite code number PG30. Kindly switch on your camera. We will be presenting your video now. Thank you.
Good afternoon, everyone. The title of my study is that exploring the theoretical potential of gentian violet in the treatment of oral cancer. The next one is background. Oral squamous cell carcinoma is a major global health issue with a common risk factor like tobacco, alcohol, and human papillomavirus. And current treatment face challenges such as toxicity and resistance. This highlighting the need for novel therapies. Here we extensively studied the role of P53 protein encoded by T53 gene. It's crucial for regulating cell cycle arrest, apoptosis, and DNA repair. And it is this regulation in cancer cell promote tumor progression and therapy resistance and metastasis. Also, we studied we investigate the role of gentian violet for its potential to restore P15 activity, suppress angiogenesis, and induce cancer cell death in chemical carcinogen-induced tongue cancer model in rats. The next one is rationale. The basic purpose of using gentian violet in our study is that gentian violet inhibit the conversion of normal P53 into mutant P53, which is responsible for tumor generation and progression. By inhibiting AKT phosphorylation, gentian violet inhibit the conversion of normal into mutant one. Due to which it causes the anti-cancer anti-cancer activity to inhibit by inhibiting P53 activity. Also improve and restore P53 transcriptional activity. And it's also inhibit NF-kappa B signaling pathway by inhibiting AKT phosphorylation. NF-kappa B is a pro-inflammatory mediator which is activated through PI3K AKT pathway. Due to AKT inhibition, due to the AKT phosphorylation by gentian violets, it's inhibit the NF-kappa B signaling pathway. Additionally, GV exhibit anti-angiogenic property, preventing tumor blood vessel formation and restoring P53 transcription activity. Also, it's modulate key oncogenic pathways such as AMG2, NADPH, and NOX genes pathway. NOX gene and NADP oxidase pathways very important in case of cancer because it may cause generation of ROS reactive oxygen species which may cause DNA damage and aberrant activity of DNA. And over the gentian violets is having inhibitory activity on these following pathways. The next one is aim to evaluate the anti-cancer effect activity of gentian violet. And the objective of study is to establish and validate the 4NQO model in spec dolly rats, and also investigate the molecular mechanism of gentian violet. The next one is methodology. The first part is establishment of 4NQO rodent model. 4NQO is nothing but a 4-nitroquinoline oxide. After treatment, it converts into active form, which is 4-hydroxyquinoline due to the generate the ROS reactive oxygen species due to conversion of NADPH to NAD, which may cause DNA damage. Also, the active form and further convert into serine AMP enzyme complex by TNA synthetase enzyme. This may cause the substitution of guanine and pyrimidine to the adenine which cause aberrant activity of DNA and also dysregulation of transcription and translation activity of DNA, which is important for cancer progression and proliferation. Next part is grouping. We included 5 groups having normal disease control, each group containing 7 animals. The third part is in vivo animal model. We mainly use 16 weeks model, starting carcinogen treatment, mixing with drinking water from first week, 16 weeks, and treatment and standard given starting from 12 weeks up to 16 weeks, and after the 16 weeks sacrification has been done. Next part is result and data analysis. The first part is food and water intake. The following figure represent the food intake of all animal from the beginning of the study and the ending of the study, which is 16th week. And the following figure represented the food and water consumption decreased by the end of the 16 weeks in all animals except the control. This attributed to the presence of oral lessons, inflammation, reduction in the metabolic rate, and weight loss due to the tumor generation inside the oral. It may cause reduction of the oral and water and food generation, food induction or food intake. Sorry. The next one is weight of tongue. The following figure represent the weight, absolute and relative weight of the tongue, and the following figure represent that there is a significant difference in the absolute and the relative weight of the tongue between the normal and disease one and treatment group. This suggests that lessons have contributed to significant rise in the weight of the tongue due to the induction of cancer. There is an increase in the weight of the tongue. Next one, ELISA, in which the protein activation have been studied. We can show that after the treatment T1, there is increase in the activity of P53 inside the body and increase the concentration as compared to standard one. So we can compare with the treatment with standard, and we can show that the old drug, which is T1, improved the P53 and restored its function more than the standard one. Next part is morphological analysis of tongue. In this, we included normal disease and standard one, in which treatment and disease induction are included. Next part is histopathological finding. It is very important for our study. The first one, A and B figure represented there is a first layer of the skin with stratified squamous keratinized epithelium. The next one is connective tissue, and beneath it is skeletal muscles. In case when disease are given, we can show that there is an increase in hyperplasia and dysplasia to the connective tissue and the stratified squamous cells. Also, it is there is also this hyperplasia in the skeletal muscles. But after the treatment, after treatment and standard are given, we can show that they effectively decrease the hyperplasia and dysplasia in both of cases. Also, we can compare the standard one with the treatment one, both of showing more higher activity. Next, with epithelial atypia index. By using morphological finding, we can score this following index. This index indicate presence of dysplasia. We can show that the T1 or treatment T1 showing lesser dysplasia as compared to standard one. By following finding, we also can conclude that the treatment T1 having higher activity on P53 and decrease the dysplasia more compared to the standard one. And having or treatment having more activity than the standard one. Here we conclude that P53 is key target for treating cancer changes induced by the 4NK model, and GB demonstrating significant anti-cancer effect compared to the disease control standard one. Also, additionally, GV hold the potential for repurposing as a therapeutical agent for oral cancer, warranting further investigation. Next one is references, and thank you. Thank you, everyone.
I now invite the judges to ask their questions to the respective presenter.
Yes, thank you. But then, um, hello, am I audible?
Yes, ma'am.
Yeah. So, gentian violet has already been reported for anti-cancer activity.
Its activity has been checked in vitro model in melanoma studies, but not has been used in oral cancer model. That's why we use as a oral model, rat model.
Okay, so how did you induce the oral cancer?
By giving 4-NQO, which is a chemical carcinogen, giving through mixing with water from 1 week to 16 week. During the 12 weeks, cancer have been developed. We can observe by seeing the whitening and thickening of the epithelial layer of the tongue.
How you are giving that means the mode of administration in water or it's directly
yes.
the oral route?
Drinking water. No, drinking water.
So drinking water. Okay. So in how many days the cancer was developed in all the animals?
Up to 12 weeks.
12 weeks. Okay. And why you are particular about P53 here?
Because, ma'am, P53, in case of oral cancer, P53 is majorly responsible. And oral drug, which is gentian violates, having a higher activity toward the P53 as compared to other mechanism. That's why we extensively choose P53, and P53 having two action. Which one is for tumor protective, and second one is tumor generation. Due to it is conversion from normal one to mutant one. That's why we choose P53 because it's also control various mechanisms such as apoptosis, angiogenesis, and other pathways. That's why we choose P53 mainly.
No, this is NQO induced oral cancer. Whether reports are available that NQO induced oral cancer P53 upregulation is observed?
In case of NQO, yes, ma'am. There is a research paper here. We can see that the NQO also increase the P53, and there also a protein analysis have been done where we can see that by inducing cancer through NQO model, there also increase in the P53 level.
Okay, you have done ELISA method to assess the P53 or just method? ELISA method.
ELISA.
What's the result? What's the
it is indicate that the oral drug having higher activity to restore the function of P53 and increase the concentration of P53 in the mice, which we have treated with 5 milligram per kg. Which is when we compare it with the oral standard one, oral drug having higher activity or higher generation of higher concentration of P53 in rats.
Now, in the treatment, what was the level of P53 in the treated one and in the control one?
Ma'am, actual level I don't know, but there is a 30 across 30, but I don't. I'm skipping the mind with the actual level.
Okay. Thank you.
Do the judges have any further questions?
Now we move on to the last presentation, PG14. Kindly switch on your camera. We will be presenting your video now.
Good morning, everyone. Our project title is Cardioprotective Effect of Pyrrolidone in Experimental Cardiac Hypertrophy via the inhibition of bromodomain containing protein 4. Cardiac hypertrophy is a condition characterized by the abnormal enlargement and thickening of heart muscle, often resulting from chronic conditions such as hypertension or heart disease. Upregulation of BRD4 protein expression has been observed in the failing heart model. BRD4 is the epigenetic reader which is responsible for inhibition of oxidative and inflammatory genes. Our proposed drug NMP, that is N-methyl-2-pyrrolidone, bind to BRD4 and inhibit its further activity. Our project aim is to investigate the effect of NMP in isoprenaline-induced cardiac hypertrophy in rat. Objectives are to evaluate the efficacy of NMP at different concentrations on isoproterenol-induced animal model of cardiac hypertrophy and to evaluate the anti-inflammatory and oxidative activity of NMP in cardiac hypertrophy. Our model is of 14 days, from which the first week is for acclimatization of animals. Day 0 to day 14, we simultaneously administered the isoproterenol and treatment for 14 days. On day 15, there is evaluation of parameters followed by sacrifice. Then following are the results. First is the effect of NMP on cardiac hypertrophy index. Cardiac hypertrophy is the ratio of heart weight to the body weight, in which we observe the significant decrease in the treatment group as compared with the disease group. Then the NMP treatment improves ECG pattern, in which there are different groups. As we seen, the treatment 3, that is higher concentration of NMP ECG, is approximately similar to positive control, that is metoprolol. Then the NMP treatment prevents expression of cardiac hypertrophy markers. There are several markers which are LDH, CKNAC, CKMB, in which there is significant decrease in the treatment group as compared with disease group. Then NMP effect on oxidative stress. It is the oxidative markers like MDA, catalase, and SOD. MDA is the oxidant, while catalase and SOD are the antioxidant. In MDA, we shown there is significant decrease in the treatment group as compared with ISO, that is disease group. In catalase and SOD, there is significant increase in the treatment group as compared to disease group, that is ISO. Then the mechanistic parameters, that is NMP inhibits BRD4 and NF-kappa B. BRD4 is the main mechanistic parameter. The NMP significantly decrease the BRD4 in treatment, while also the NF-kappa B levels are also decreased in the treatment group. Then these are the histopathological studies, in which first row indicates the fibrosis and collagen deposition, which is stained by Mason trichome staining, in which the ISO group, that is disease group, shows severe fibrosis with collagen deposition, and the treatment group, higher concentration of treatment group, shows mild fibrosis with very less collagen deposition. Then the second row indicates inflammatory cells and edema, which is stained by hematoxylin and eosin stain, in which the ISO, that is disease group, shows high inflammatory cells or high edema as compared to higher dose of treatment, that is 500 mg per kg. NMP shows mild edema with less inflammatory cells. Here we are concluding that our study demonstrates the potential of NMP in alleviating cardiac hypertrophy by isoproterenol in rat. NMP treatment improve cardiac parameters, reduce oxidative stress, and decrease inflammation. The mechanism of action was linked to the inhibition of BRD4 and NF-kappa B pathway, suggesting NMP's potential as a therapeutic agent for cardiac hypertrophy. These are the references. Thank you.
I now request Charya to ask the question.
Okay. Could you please let me know what's your title? NMP.
Yeah, ma'am. My drug name, N-methyl 2-pyrrolidone.
So, okay. So why you have to, it's whether it was synthesized?
No, ma'am, it is obtained from vendor.
No,
Purchased from
what's
vendor.
the, okay, what's the rationale for using that NMP for cardioprotective activity?
Yeah, ma'am. NMP is bind to BRD4, and BRD4 are responsible to inhibit the inflammatory or oxidative stress which are generated in cardiac hypertrophy. So it is our rationale because in cardiac hypertrophy, BRD4 level are increases and NMP is reduced that levels.
Okay. So what is the current? This is a chemical, right? It is.
Yeah.
What's the other?
Yes, ma'am. It is the solvent which are used in clinical trial for some diseases.
So that's what for what purpose?
Used in melanoma or clinical trials.
Okay.
Yeah.
Okay. So it can also be used. That's what it can also be used for cancer, right?
Yes.
So you are evaluating for its cardioprotective activity based on the target.
Yes. Yes.
Okay. So how did you assess this BRD4 expression by Western blot or?
No, ma'am, by ELISA.
Okay. What was the dose of isoproterenol?
Yeah, ma'am.
Inducing agent to induce cardiotoxicity.
5 mg isoproterenol.
5 mg.
For 14 days. It is administered daily for 14 days. My study is of 14 days.
5 mg for 14 days.
Yes.
Okay. And this NMP treatment is for how many days? What is the dose and what's the dose of NMP and what's the duration of treatment of NMP?
NMP is also administered daily for 14 days and the dose is 3 doses are there, 100, 200 and 500 mg per kg.
On what basis you decided the dose?
Ma'am, according to literature.
Literature based on cancer studies or?
Yes, ma'am. It is cancer study based.
200, 300
and we are obtained the results on that doses.
Okay. Thank you.
Thank you, ma'am.
Have you looked at the toxicity profile of this molecule?
No, ma'am, it is not that much toxic. It is water. It is water soluble drug, and we find some adverse effect in clinical trials, which are means nausea, vomiting, etc. Minor side effects. Not much that toxic. Or it is toxic in pregnant women. Means it is not given to pregnant women.
So then can this kind of a molecule be taken ahead for even testing? What is its PK?
PK.
See, if it's very polar and if it's very non-polar, then it will fail in biodistribution, no?
It's polar, ma'am.
Exactly. So when a molecule is polar or non-polar, it will fail in biodistribution, isn't it? Right. So, what was the strong rationale that you had for picking this molecule?
Ma'am, my main focus on BRD4, which is the main mechanistic parameter of cardiac hypertrophy. And the drug is acting on that parameter and decrease the level of BRD4.
Okay. Okay, fine.
You, ma'am.
Do the judges have any other questions they would like to ask?
No, that's it.
We thank the judges for their valuable questions. Dear judges, please make sure you have filled the scorecards.
As we approach the end of this session, I would request the judges to say a few words about the experience of this event.
Ma'am, you can start because we are the host.
So I would take this opportunity to thank all the judges for spending the entire day with us and judging this all entries and appreciating our students and our means the all participants students presentation and their skill. We sincerely thank your time, Dr. Manjula. I know you're very busy. In spite of that, you have spent the entire day with us. Thank you very much, Dr. Radhika, Dr. Manjula, Dr. Rajkumar. Thank you very much. We really sincerely appreciate your time and efforts which you have spent today for this competition, for the event. And I'm sure your contribution is going to go a long way in learning journey of ours or students all who have participated in this. So thank you once again, and thank you students for handling this event such wonderfully. Over to you.
Thank you so much, madam. At the outset, I am very thankful to you for giving us an opportunity to be the part of this fifth Student Research Congress. In fact, I'm very much happy to be the part of this. It's students, as you rightly mentioned, the research work, what they have done is at the PG level, it is like a PhD work. So really it is all, I don't know how others are also have participated, and you have done a very excellently, you have scrutinized, and all the presentations are excellent research work, madam, really. And the methodology, the rationale, and the results, all each and every aspect, the protocol designing, each and everything is excellently well designed research work. And even the presentation also, it's excellent, madam, because whatever the work they do it's very difficult to present in such a small duration. So all the students have presented their research work, the entire study, in a given span of time. That's really very great thing. And it's all the newer concepts also they have incorporated. It's at the PG level, really, it's a wonderful experience to me, madam, in fact. Thank you very much for giving me the opportunity. It was very good learning experience for me as well, madam. Thank you so much.
Thank you very much, Dr. Manjula.
Would any other judge like to speak a few words?
I think Dr. Manjula has spoken on behalf of all of us. So she really summed it up well. Good, very good research work, good methodology, and finally good presentations also. So all these go a long way for your future when you are parts of the research teams in pharma companies. Good.
So that's what I always say, that winning is not important, participation is important, because when you participate, you also listen to all the presentations, and that's where it's a learning journey starts. So that learning opportunity itself is a winning. So all of you are winner. Over to you, students.
Thank you for your kind words. On behalf of the organizing team, it is my privilege to deliver the word of thanks on this special occasion. First and foremost, I extend my heartfelt gratitude and deepest respect to our distinguished judges. Your invaluable time, insightful feedback, and commendable knowledge have not only graced this event, but also enriched our learning. Thank you for your generous contributions to the success of this event. We would like to present certificates of appreciation to our judges. Dr. Rajkumar,
Just coming early.
Dr. Manjula, and Dr. Rahida.
A big round of applause and sincere thanks go to all the participants who have truly made this event memorable with their enthusiasm, creativity, and dedication. Your efforts are the heart of this competition, and we are incredibly proud of each one of you. Lastly, I would like to thank everyone present here today. Your support and presence has been instrumental in making this event a grand success. Thank you for Thank you for taking the time to be with us and contributing to this inspiring experience. Once again, thank you all and see you tomorrow.
Thank you, everyone. Thank you, all the participants. And see you tomorrow, everyone, for PhD and innovation entries. Thank you.